वो मुझे शेड्यूल के अकॉर्डिंग सेकंड डे के स्पीकर्स के एब्स्ट्रैक्ट मेल कर दो, रिपोर्ट के लिए चाहिए था। ओके।
डॉक्टर साहब आप म्यूट नहीं है, आवाज आ रही है बैकग्राउंड में।
यस सर, यस सर। म्यूट है सर, म्यूट। Good afternoon, sir.
Good afternoon,
Good
sir.
afternoon. Good afternoon. Very nice.
Nice to see you, sir.
Yeah, I too.
Good afternoon, sir.
Good afternoon.
We have 3 minutes to start.
Yeah.
Yeah.
Good.
Dr. Vijay?
Hello.
Hello.
Ma'am, good to go. Start.
Okay, Dr. Sabati. A very warm good afternoon to all of you. To proceed with the 3-day international seminar, international conference, I, Dr. Pragati Mishra, co-organizing secretary of this international conference, on behalf of the Department of Molecular and Cellular Engineering and International Academy of Physical Sciences, Prayagraj, first of all, extend my most sincere thanks to the Almighty God for making this event a resounding success. This has been an absolutely amazing and enlightening online international conference. Dear participants, hope you all are enjoying and enlightening by the lectures given by different speakers. Today we have 4 speakers or experts in our different fields of biotechnology, biochemistry, and bioinformatics who will share their views and exchange knowledge. Now, before we start the technical session, first of all, I would like to introduce the chairperson of the 3rd day of International Conference, Professor Dr. P.W. Ramteke. Professor P.W. Ramteke is currently ex-professor emeritus, Faculty of Science at Sam Higginbottom University of Agriculture, Technology and Sciences, Prayagraj. He has served as Dean Postgraduate Studies and Head of the Department of Biological Sciences, SHUATS. Professor Ramteke did his PhD in microbiology in 1993. He edited many books and published large number, more than 375 research papers, book chapters, and popular articles. He is having more than 5,000 citations. He is in the editorial board of many reputed international and national journals. Professor Ramteke has more than 36 years of teaching and research experience in microbiology, biotechnology, and nanotechnology, and contributed significantly towards development of microbe-based eco-friendly processes to utilize agriculture waste for production of biofuel and industrial important enzymes, sustainable crop production, biodegradation, and detoxification of environmental contaminants, and biorecovery of heavy metals from agriculture run-off waste waters. With this brief introduction of such an eminent personality, I would like to invite Professor P.W. Ramteke for chairing the 3rd day of this international conference, CONIAPS 2006. Now it's over to Ramteke, sir.
Thank you very much, Dr. Pragati, for nice introduction. I'm audible?
Yes, sir. Yes, sir.
Yeah. Very good morning to all the participants and presenters for this session, 3rd day session. Our first speaker today is Dr. Dhruva Kumar. And before I invite him, I would like to give his a short introduction. Dr. Dhruva Kumar completed his PhD in cellular, molecular and industrial biology from the University of Bologna, Italy, under Indo-Italian Government Fellowship in 2012. After completion of his PhD, he received 4.5 years of postdoctoral training from the University of Kansas Medical Center, Kansas City, USA. There he worked on translational cancer research. During his postdoctoral training, he worked on several cancers, including prostate, pancreatic, breast and head and neck cancer. First 2.5 years of his postdoctoral training was focused on understanding the molecular mechanisms of the regulation of autophagy and apoptosis in cancer stem cells. In addition, Dr. Kumar also worked on intra-tumor mutational heterogeneity in head and neck squamous cell carcinoma, developed an in vitro model for radiation-induced fibrosis in head and neck cancer, molecular mechanisms of combination of naturally derived chemopreventive agents like curcumin in association with other anti-cancer compounds. Apart from his research, he is involved in teaching courses related to cancer biology, stem cell biology, molecular medicine, and bioinformatics. I invite Dr. Dhruva Kumar to present his talk. Dr. Dhruva Kumar.
Yeah, thank you very much. Thanks a lot, Professor Ramateke. And I would like to thank the organizing committee, Dr. Vijay, other organizing members of this CONIAPS conference. And it gives me immense pleasure to talk and discuss about my work here on this platform. And I'm a lifetime member of this CONIAPS. I'm also a young scientist awardees by this CONIAPS. When I was doing my PhD in 2011, I got awarded for my paperwork. So it's really a pleasure for me to talk on a same platform here as a speaker and to discuss about my research work, current research work. So thank you very much, Dr. Vijay, again, and the organizing committee. So with your permission, may I start sharing PPT, my presentation, Dr. Vijay?
Yeah, sure. Yes, sure. Please.
Yeah. Thank you. So I will be talking about the signaling regulations as you have mentioned that I'm working on autophagy, metabolism, cancer cell, cancer stem cells. So I will be talking about the signaling regulations and mutational heterogeneity in cancer and cancer stem cells. And specifically, the signaling crosstalk. The two major pathways are there, or the regulations you can say. The one is autophagy and second one is metabolism. So these two regulations are highly altered in cancer and cancer stem cells. And there are lots of things, including cancer prevention, cancer therapeutics, can be done through targeting these autophagy and signaling, metabolic signaling pathways. So if you see the cancer geographical distribution of cancer in India, starting from the Jammu Kashmir to the Kanyakumari, the most common cancer type, if you see here in this picture, it's a oral cancer. It's a head and neck cancer in men, and in women it's a breast cancer. Now the question is, what are the factors, how it is associated with the cancer progression, and how it is initiated. There are lots of things, studies are being done on that. So I will be talking about, if you understand the signaling pathways, the signaling regulations of these cancer and cancer stem cells, then you may able to manage or able to develop a therapeutic approach for that. So in my lab, we are working on different signaling pathways. We are targeting PI3 kinase, mTOR, AMPK, c-MET, FGFR. All these signaling pathways we are studying in autophagy as well as metabolic regulations. We are also trying to understand how these genes are responsible for cell proliferation, metabolism, metastasis, drug resistance, and stemness. Another work that we are doing is HPV associated human papilloma virus. It's a virus that mainly associated with cervix cancer. But recently, there are some reports that they have demonstrated that, that from the clinical data, that this HPV association is also associated with breast cancer, oral cancer. Now people are claiming that it's associated with pancreatic cancer as well. So the signaling regulations in HPV in cancer and cancer stem cells specifically, in sense of metabolism, the HPV negative versus HPV positive is totally different. So all these things we are studying in our lab currently. So in this today's lecture, I will be mainly talking about head and neck cancer, the signaling regulations in head and neck cancer, as well as the tumor heterogeneity in head and neck cancer, and how that heterogeneity, understanding of heterogeneity, can be used to develop personalized therapeutic approach for cancer. So talking about the current status, the current scenario of head and neck cancer, it's the most common cancer in men in India. and it's the 6th and 5th most common cancer in worldwide. But the problem is, if you see the 5-year survival rate of this cancer is less than 50%. So not many cancer patients with a head and neck cancer, they survive for more than 5 years. And the factors associated with this cancer is tobacco, alcohol use, HPV, as I mentioned. So you see, this cancer appears mainly in the region of neck, oral cavity, tongue, lips, larynx, and pharynx. And if one is a little bit vigilant, then this cancer can be cured, can be prevented easily. Now, if you see the solid tumor, any kind of solid tumor, it's not only cancer cells. There are group of cells that participate in cancer progression. And those cells are immune cells, fibroblast cells, endothelial cells that we call blood vessel cells, and tumor-associated fibroblast cells or cancer-associated fibroblast cells. Now the question is, what this the tumor microenvironment, the other cells, they do in tumor progression. And if they do, then what kind of signaling pathways are regulated and how it is regulated? As I mentioned earlier, the autophagy and cancer metabolism, these two processes are highly altered. And these two processes are the main, the cancer progression regulatory process. So in order to kill the cancer cells or cancer stem cells, if you are targeting metabolism, because metabolism is the energy source for cancer cells, and autophagy, through which they develop the drug resistance, then you will be able to, at some extent, kill the cancer cells and cancer stem cells, and to solve the drug resistance problem as well. So the normal, the process of tumor, the general process of tumor initiation is, normally it happens with the genomic instability. As you can see here, there are several factors: carcinogens, viruses, dietary factors, smoking, drinking, radiations. All these factors are responsible for genomic instability in our cells. When our cells are genomic unstable, then they do not follow the cell cycle checkpoints. Cell cycle checkpoints that regulate the cell growth. And what happens? As a result, you see lots of cancer cell growth. And that cells, they become like a tumor. And what happens? These cells, when they grow in a tissue environment, tissue microenvironment, they secrete lots of growth factors in their neighboring tissues. And these growth factors, again, they promote fibroblast cells, which are present in connective tissues, as a cancerous, that we call tumor-associated fibroblast or cancer-associated fibroblast. So this is how, in tumor microenvironment, these cancer cells and cancer-associated fibroblast cells, they help each other in cancer progression and cancer growth. So our idea is to just to break this bridge, as you can see here. So our idea is to understand the signaling pathways which are regulated in cancer cells and cancer-associated fibroblast cells, and to break this bridge that help cancer cells and cancer fibroblast cells to grow. So this is the histological section of oral tumor. As you can see, the major area of this oral tumor, it has a fibroblast cells, which is stained with alpha SMA protein. So as I mentioned earlier, our current approach is to understand signaling pathways as well as the mutational heterogeneity in head and neck cancer and cancer stem cells specifically. So what we do, we collect conditioned media from fibroblast cells, and then we use that conditioned media to treat, to see the effect of tumor microenvironment or cancer-associated fibroblast cells on cancer cells. So green color represents the cancer cells, and this one, the red staining, is the cancer-associated fibroblast cells. So we collect media from vice versa, from cancer-associated fibroblast cells as well as from cancer cells, and then we treat these two cell types in vitro culture to understand their cell proliferation, migration, invasion, and the gene regulations with and without these cells, how cancer cells behave. And then we perform all these assays to understand the signaling regulations. So as I mentioned earlier, it is very important in case of therapeutics development, what kind of target you are identifying, what kind of regulatory proteins you are identifying in cancer cells or the other cells which are responsible for cancer growth and migration or progression. So what we did, we have identified two the targets. One is c-MET, that is called receptor tyrosine kinase, the c-MET receptor in cancer cells, head and neck cancer cells. Another target we identified in cancer-associated fibroblast cells, that is FGFR. And we'll discuss how these proteins, the FGFR signal, regulates the cancer-associated fibroblast cell progression, and this c-MET regulates this cancer cell progression through metabolic activation. And these two signaling pathways, they talk to each other between cancer cells and cancer-associated fibroblast cells, and they help in tumor growth and tumor progression. So our main goal is to block this signaling regulation in c-MET in cancer cells and FGFR regulation in cancer-associated fibroblast cells to inhibit the cancer progression. The normal process of metabolism goes through this mitochondrial respiration, but in cancer cells it goes through lactate. So that's a simple difference in cancer cells and normal cells. So you can measure metabolic activities through this instrument that we call seahorse plate reader. So we have used this plate reader to measure glycolysis as well as mitochondrial respiration in cancer cells and tumor-associated fibroblast cells. So as you can see here, glycolysis is high in cancer cells, whereas mitochondrial respiration is high in tumor-associated fibroblast cells. So these two cell types, they have a different metabolic regulations. Okay, so we have inhibited glycolysis in cancer cells and mitochondrial respiration in tumor-associated fibroblast cells to stop the cancer growth. So this is another experiment that we measured the end product of glycolysis, or the product that utilize cells, okay, for their cell proliferation. This is the typical graph that you will see, the data that we collect from glycolysis. And as you can see here, in control without any treatment, okay, the cancer cells, they are growing, they are doing metabolism, but when we have treated these cancer cells, we saw significant difference in their growth. So the red color indicates cancer cells with conditioned media isolated from TAF, and this is the in a normal conditioned media. So what we did, we inhibited this the c-MET receptor with a specific inhibitor, and then we measured glycolysis. So this glycolysis was inhibited with this inhibitor that you can see here, and that was directly proportional to the cell proliferation as well. So that indicates that when you block this glycolysis, this c-MET regulation in cancer cells, you can stop the cancer cell growth and proliferation. And further we measured the hexokinase enzyme which is responsible for the conversion of glucose to glucose 6-phosphate. As you can see here, with this inhibitor, we saw decrease in hexokinase expression. Yeah. So another experiment what we did, we co-cultured cancer cells and cancer-associated fibroblasts together to measure a protein called monocarbogylate transporter. This transporter transports lactate from inside to outside in a cellular environment. So this expression was also high when we co-cultured. It means cancer cells, they perform glycolysis more when they come together with the cancer-associated fibroblast. So with the inhibition of this c-Met, as I have mentioned earlier, we have identified two main signaling pathways in cancer cells and cancer-associated fibroblasts. One c-Met in cancer cells and FGFR in cancer-associated fibroblast cells. So our goal was to block this protein in cancer cells and FGFR in TAPs. So here, this ligand actually, another story, it's a very interesting story. To activate this protein, cells get ligand from cancer-associated fibroblast cells. So what we did, we inhibited c-Met with a specific inhibitor that we called, So in a cancer cell progression, when SGF binds to the c-Met, it regulates glycolysis, hexokinase expression, as well as the expression of basic fibroblast growth factor. So you see, the very interesting thing, the ligand for this c-Met is coming from cancer-associated fibroblast cells. And here, the ligand for this FGFR is coming from cancer cells. So these two signaling pathways, they talk to each other. Okay, the cancer cells, they talk to each other through these signaling pathways. The second part of this, the metabolic story, is we measured mitochondrial respiration in cancer-associated fibroblast cells. So this red color indicates the normal mitochondrial respiration. When we inhibited this mitochondrial respiration with AJD, which is a specific inhibitor for FGFR, as you can see here, the respiration is very low when we treated these cells with the specific inhibitor. Other parameters we have measured, but just coming to this, so when we treated these cancer-associated fibroblast cells, excuse me, with the specific inhibitor of FGFR, that is called AZT4547, we saw the decrease in that protein expression. It tells that the FGFR regulates the MAP kinase, because this AZD4547 is a FGFR inhibitor, not MAP kinase inhibitor. So what we are doing, we have done here, we have inhibited FGFR here, not MAP kinase, but FGFR inhibition decreased the expression of MAP kinase. And that MAP kinase further, decrease the expression of HGF. So this FGFR regulates this MAP kinase as well as HGF expression, both in cancer-associated fibroblast cells. So what we have seen so far, this HGF regulates c-Met and this BFGF regulates FGFR, which is a specific ligand for FGFR. And this FGFR regulates MAP kinase, oxidative phosphorylation, as well as HGF secretion from cancer-associated fibroblast cells. Further, we did this experiment on a mice model, and we found that, the mice wearing tumor treated with c-Met inhibitor as well as AGD inhibitor that is HJD, that is FGFR inhibitor together, and we saw that significant decrease in their the tumor. So that is very interesting finding we saw. So at the end, what you see, the c-MET is activated by this HGF, and when c-MET gets activated, it regulates glycolysis as well as basic fibroblast growth factor secretion from cancer cells. And this basic fibroblast growth factor activates FGFR, and that FGFR activation regulates MAP kinase as well as oxidative phosphorylation that we call mitochondrial respiration in cancer-associated fibroblast cells. And this MAP kinase activation, oxidative phosphorylation, all together activates HGF production from this particular cancer-associated fibroblast cells. And together, these c-MET signaling and FGFR signaling, these two signaling pathways, they talk to each other to promote cancer or tumor growth. So what we did, we used a specific inhibitor for c-MET, and we inhibited all these signaling pathways. Then we used AZD4547, a specific inhibitor against FGFR, and we blocked oxidative phosphorylation, MAP kinase, as well as HGF secretion from TAPs. And together, what we saw, the reduction in tumor growth and tumor progression. So this was our specific targeting specific signaling pathways in cancer cells and cancer-associated fibroblast cells to inhibit tumor progression as well as tumor growth. So to conclude this part, head and neck cancer cells are more glycolytic than tumor-associated fibroblast cells. TAF induces glycolysis in cancer cells through HGF and c-MET axis. Head and neck cancer cells increases mitochondrial respiration in TAPs through basic fibroblast growth factor and fibroblast growth factor receptor. And targeting this HGF c-MET and BFGF FGFR signaling pathways in head and neck cancer, you can reduce the tumor growth. So some of the research article came out from this work. Now talking about mutational heterogeneity. Nowadays, the biggest problem that we are facing is cancer drug resistance. And that cancer drug resistance develops through mutational heterogeneity. Even you see, when you analyze tumor samples from the same stage, even from the same family, you will find two tumors are completely different in the sense of mutation, the gene expression, protein profiling, and that kind of difference we call mutational, the heterogeneity. The two types of heterogeneity, is there intratumoral and intertumor heterogeneity.
Hello, Dr. Dhruv
Yeah.
Kumar. Hello, Dr. Dhruv Kumar. I think your time is over. You can conclude.
Yeah, just give me 2 minutes.
You have a 25 minutes time.
Okay, fine. Yeah. So what you see, the inter and intertumoral mutational heterogeneity. So in this work, what we have identified that the difference in single nucleotide variation in the different patient samples of head and neck cancer, as well as the difference in copy number variation. So we have identified different mutations, okay, that's the missense mutations in a specific tumor from the larynx portion, okay. So the point is this mutational heterogeneity that we have identified in a cancer patient, head and neck cancer patient, that is directly associated with the toxins that we take, like the mutagens. In case of larynx, you see here, it's a highly heterogeneous because when people smoke, the smoke comes through the oral cavity and it affects the laryngeal region. And this is the reason why this sample has lots of heterogeneity. Okay. We identified a specific mutation in P53. And to conclude this, the degree of mutational heterogeneity depends on the factor that we take as a carcinogen. Okay. So this work we published in OncoTarget. Now the important point here, just will take 30 seconds. If you understand the mutational heterogeneity, signaling regulations in a specific tumor or a specific cancer, you can easily design a drug molecule. You can easily target those specific signaling pathways in a specific cancer patient. And that is called personalized cancer therapy. So nowadays, most of the, the cancer institutes, they are focusing on personalized cancer therapy. And it's a highly successful therapeutic approach. So with this, I conclude this lecture. And these are the lab members and collaborators. With their support, we have done this work. And now I am open for question. So thank you.
Yeah, thank you, Dr. Dhruv Kumar, for such a good talk on blocking of specific signaling pathways in the cancer cells. Maybe we have one question if somebody is interested to ask. Or you are always free to approach Dr. Dhruv Kumar and get your queries sorted out.
Yeah, I have given my email there. So anyone, if interested in asking any question, they can write me an email.
Yeah.
Yeah.
Dr. Dhruv Kumar, it was such a wonderful talk. Thank you very much.
Thank you. Thank you.
Okay. Yeah. Our next speaker is Professor Just Andre. Before I invite him for his talk, I have a privilege to introduce to you him. Dr. Just Andre is a professor at the Faculty of Bioscience and Aquaculture at Nord University in Norway. Where here he has established the landscape genomics group, and the aim of this group is to understand local adaptation and adaptive potential in natural, human disturbed and exploited populations across large and ecological diverse landscapes. Previously, he had worked as a postdoc fellow at the University of Leuven in Belgium, University of Basel in Switzerland, and McGill University in Canada. His main research areas include population genetics and evolutionary ecology. He is particularly interested in local adaptation of ecological speciation, adaptive radiation, host-parasite interactions, and eco-evolutionary dynamics. At Nord University, Professor Andre is teaching genomics and bioinformatics and biological data analysis. Thank you very much, Dr. Andre, for joining us. And now I invite you to deliver your talk.
Thank you, Professor Ramteke. Can you hear me well?
Doctor, yes, it's audible.
Okay.
Yes, we can hear you.
Okay, I'm sharing my screen, I hope now. Is that correct?
Yes, yes.
You can see the slides, okay?
Yes.
Okay.
Yes, we can see.
Okay. Thanks a lot for the nice introduction and the invitation to CONIAPS. I'm very happy to present some of my work in the field of landscape genomics. And I'll start with the Sustainable Development Goals. And the last 2 days, we had many interesting talks covering some of these goals. We have been dealing with wastewater treatment, for instance, environmental resilience, also the healthcare system, microbial diversity, the quality of life in general, COVID, of course. And but today I would like to cover Sustainable Development Goals 13, 14, and 15, which deal more with climate change and biodiversity. So, as you all might know, we are dealing with a climate change crisis, which has anthropogenic causes relating to the CO2 levels in the atmosphere, which are the highest in 440,000 years. And so there's, and this has to do with industrial activity, mainly the production of greenhouse gases. And then there's another crisis on the planet, which is the biodiversity crisis, and which currently is referred to as the sixth mass extinction. So we have all the biodiversity on Earth, plants and animals. But for instance, from all the terrestrial diversity, 31% is in decline. And 1/8 and 1/4, 1/8 of the birds, 1/4 of the mammals, 1/3 of amphibians, and 6/7 of marine turtles are at threat of extinction. And also 50% of Earth's forests are cleared with annual loss of more than 130,000 square miles. And we've lost 75% of genetic diversity of agricultural crops, and 75% of the world's fisheries are fully or overexploited. And in reality, the true picture is much worse, since the conservation status of most species is poorly known. Now, what are the causes of biodiversity loss in order of importance? That's changes in land use and sea use, exploitation of organisms, climate change, pollution, and invasive species. So you see climate change is there in this list. And so the climate change and biodiversity crisis interact with each other. This is illustrated here. These are some of the world problems. They're actually surrounding the overpopulation, the human overpopulation. But here you have climate change and biodiversity next to each other. Also at the political level or at the policy level, there's two panels, the IPCC, Intergovernmental Panel of Climate Change, and the IPBES, the Intergovernmental Panel on Biodiversity and Ecosystem Services, which work closely together. And biodiversity supports crucial ecosystem services. Here you see a long list of them. But here in red, I have marked the one which are supported by, which are actually relating to climate change. So the biodiversity crisis may accelerate climate change effects because they support these processes. And therefore, if you have climate change, you have biodiversity loss, therefore these processes will be less supported, which could lead to more climate change and therefore to more biodiversity loss and so on. So biodiversity problems threaten the continued existence of our own species and the future of the biosphere. So now, are there solutions to this problem? Greta Thunberg, which is a climate change activist, refers to climate change as a crisis. And she says that we should to solve the crisis, we should treat it as a crisis. And here I made a comparison between cancer biology and conservation biology. As Professor Kumar pointed out, cancer biology has several factors. So it's a very complex discipline, but it's needed to treat the crisis of cancer. And so the patient in this case are the humans. For climate change and biodiversity, the patient is not humans, it's the earth. And the science of conservation biology is also a crisis science or a crisis discipline supported by many subdisciplines. And today I would like to focus on this part of the disciplines: population biology, population genetics, ecology, and genetics. So what knowledge can we use to help conserving biodiversity? Now, starting from ecological theory, upon environmental change, a local population of a species has the following options. It can tolerate the environmental change, it can try to avoid the environmental change, or it can try to adapt to this environmental change. However, there's a fourth option. If any of those three are not possible, then the local population will go extinct. But conditions on the planet get more and more extreme, so it's not always possible to tolerate those conditions. Movement or migration is not always possible, so it's not always possible to avoid the environmental change. And then adaptation requires plastic phenotypic responses or beneficial genetic variants or alleles. And it's on the last part I would like to focus, because this is what we define as adaptive potential. It's the adaptive potential which will tell whether a population of a species will be able to adapt to changing environmental conditions if tolerance or avoidance is not possible. So adaptive potential is the ability of species or populations to respond to environmental change. And this adaptive potential is to a large extent rooted in a population's genetic diversity, which I illustrate here with genes or alleles in different colors, different genetic variants. And this genetic diversity forms the basis, the basic level of genetic diversity of the ecosystem, in addition to intraspecific diversity, community and species diversity, and then ecosystem diversity. And genetic diversity is subject to major evolutionary forces in nature that control genetic diversity. First, there's selection, which targets beneficial genetic variants, as illustrated here. In a green environment, green variants are beneficial. In a blue environment, blue variants are beneficial. The second major evolutionary force is genetic drift, which leads to the random loss of genetic variants in small and isolated populations. Here you see a large population with quite some genetic variants, but when populations get really small, then some of the genetic variants will be lost. The third force is gene flow, which homogenizes the gene pool across populations. So if you have two populations, there's a certain connectivity between them, certain migration between two populations, and so they exchange genetic variants. Okay, now, how can landscape genomics be useful? Landscape genomics is a tool to understand and predict the distribution of adaptive potential across the landscape. How is it doing so? Well, it enables us to quantify genetic connectivity, for instance, to see where in the landscape we find major migration barriers. And on the other hand, it helps us to quantify adaptive divergence, which tells us how local populations will adapt to their local selective environment. And all this is done based on genotypes. So with these genotypes, we can map genetic diversity in space to infer the processes of selection, gene flow, and genetic drift. And so there's a spatial component, which is indicative for connectivity, and there's an environmental component, which is indicative for adaptation. And so we can quantify the processes in the landscape that influence the distribution of genetic diversity and the major processes, gene flow and selection. And this will help us to assess population persistence, because genetic diversity supports this adaptive potential and therefore will tell us how resilient a population will be to new challenges in the future. So landscape genomics has multiple applications that can lead to practical solutions which are applicable to a wide range of spatial scales and environments, as summarized here in major reviews and synthesis. And so it's actually also relevant to all scientific disciplines where spatial distribution of genetic diversity matters. So conservation, pest control, but also forestry, crop production, wild crop varieties, fisheries, bioindicators, and biocontrol, agriculture in general also, of course. So for the remaining of the talk, I'm going to give some examples. I'm not sure I will get through all of the examples because of time constraints, but I will start with riverscape genetics, then go to landscape genetics, lakescape genetics, and seascape genetics, demonstrating that you can apply landscape genomics to different ecosystems. For riverscape genetics, I would like to focus on stickleback fishes from the Northern Hemisphere. For landscape genetics, I'm gonna use a case study on Svalbard reindeer. For lakescape genetics, we jump to Africa for a study on Lake Tanganyika sardines. And for seascape genetics, I'm going to look to flatfish species of the North Sea. So let's start with riverscape genetics. This is a study which we conducted in 2008 in Flanders, the northern part of Belgium, where we studied this drainage, which has a lot of migration barriers, including historical water mills, but also more recent constructions such as weirs and tunnels and other human-made constructions. And in this river system, you can see the distribution of all these migration barriers, but we decided to look to the fish populations at the black dots each time upstream and downstream each side river. So at each of these dots, we collected 40 individuals of three-spined stickleback, this small fish. And we quantified genetic diversity using a small panel of microsatellite markers. So we genotyped the fish and calculated genetic diversity based on the allelic richness of this panel of microsatellite markers. And we observed that the allelic richness varies between 4.5, which is really low, up to 12. And then we mapped this genetic diversity to the river system, and we concluded that the distribution is as follows. In the upstream part of the basin, we find a lot of populations with low genetic diversity, whereas if you go more downstream, then allelic richness increases or the genetic diversity increases. And then we compared this with two geographical scenarios, a scenario where genetic diversity is shaped by river distance and another geographical scenario where genetic diversity is shaped by migration barriers. And then we verified which of those two scenarios matches best with the genetic diversity. And it turns out that the migration barriers correlate much better with allelic richness than the river distance. We're now extending this approach to multiple species from the same watershed. This work done by my PhD student in Belgium, Iyo de Flem. So what is the conclusion of this part on riverscape genetics? Is that migration barriers and more than natural factors impact the genetic diversity of riverine fish populations. And especially the most upstream populations are at risk of genetic diversity loss. So using riverscape genetics, we have identified which populations in a large watershed are the most endangered for future changes, are most at risk under future changes. Okay, I'll move on to landscape genetics. Moving much more north now to Svalbard, which is an archipelago north of Norway and Russia. Here I'm going to present a very similar study as the case of the stickleback fishes, but on Svalbard reindeer populations. And here, this map is to situate where is Svalbard, close to the North Pole. And this reindeer has been have a history of over-harvesting until 1925. After that, there was population recovery and recolonization across the archipelago. And even later, some of the populations have been reintroduced and then they naturally expanded. So that's what happened with the populations, but what happened in the meantime with the genetic diversity of these populations? That's the question we wanted to answer with a study where we collected a lot of reindeer samples, either live reindeer using blood samples or stool, or historical DNA from bone. At the same time, there's another part of the history, and that's the sea ice loss due to climate change. Here in red, you see the sea ice in 2009 on the left, and the sea ice in 2018 on the right, in the same time of the year, both mid-February. And you see a large decline of the sea ice. Now, before I come back to the sea ice, I'm going to show the results on genetic diversity, where again, in red, you have regions of high genetic diversity, and in blue, where you have regions of low genetic diversity. And it turns out that the genetic diversity is very low at the periphery. So here in the center of Svalbard, you get populations with high genetic diversity, but on the edges, the genetic diversity decreases a lot. very similar to the upstream populations in the stickleback fishes. Then some other analysis were aiming at analyzing the genetic differentiation, looking how different the populations are genetically. And here we noticed again that if we look to different scenarios, then we observed that the best geographic scenario to explain the genetic differences between populations across the archipelago is a scenario where we assume that individual reindeer, migrate over Svalbard using a combination of land and sea ice. So if we assume that the reindeer cross the ice, then actually we find the best prediction of the genetic connectivity. It actually means that migration over sea ice is the best predictor of population connectivity for these populations. If we exclude migration over sea ice, we don't find a good correlation with the observed genetic diversity. So it does imply that this reindeer use sea ice in winter to go from one location to the other. Conclusions from the study is that sea ice loss will continue to isolate reindeer populations with climate change. And therefore, conservation strategies such as assisted migration will become necessary in the future to slow down the loss of genetic diversity. So the next ecosystem are the lakes. So switching now to lakescape genetics. For this, we performed a study on Lake Tanganyika, which is in Central Africa, bordering Congo, Burundi, Zambia, and Tanzania. And this Lake Tanganyika has two very important species for the local artisanal fisheries. It's two clubits, it's Stolothrissa tanganyika and Limnothrissa myodon. We call them freshwater sardines. And this lake is pretty large. It's 670 kilometers north to south. And we sampled the lake in the north, in the middle, and in the south. And then we performed again genetic analysis, in this case using a panel of single nucleotide polymorphism markers. With these genotypes, we again assessed population structure. We made this two-dimensional visualization to test whether populations of the north, central, and south of the lake are differentiated or not. It turns out that this was not the case. Across the whole lake, the population is actually near panmictic. This means that there's very little population structure from north to south. And so what we can conclude from this study is that Stolothrissa tanganyika can be managed as one single stock among the 4 countries bordering Lake Tanganyika. This is a very important conclusion because we're talking here about 4 countries which are economically not very well developed. They have this fish stock as a major resource, and this shows that they have somehow to collaborate to manage the fish population. And the good thing is that they don't have to take into account that there are different genetically differentiated stocks, which is easier management than if we would have different stocks, each with their own genetic signature. Final ecosystem is seascape genetics, where we look to these 3 flatfish species of the North Sea. Here we did a very similar study as for Lake Tanganyika. We studied the population structure of these 3 fishes in the North Sea, where again these 3 fish species co-occur. You can catch them during the same trawl. And the 3 species differ in life history. So they differ, for instance, in the timing of spawning during the year. Now, the North Sea is not like a lake, it's much more dynamic system. In some seasons, you have this oceanic fronts, which are basically temperature salinity barriers, which form to some extent a migration barrier for larvae when they disperse. The larvae tend to stay in the same temperature salinity zone. And therefore, the duration of the spawning time can influence the genetic structure of these fishes. Now, it turns out that those 3 fish species differ quite a lot in their spawning time, and therefore they do have very different genetic structure, even though they live in the same environment. And that's what we analyzed here, again, using microsatellites we showed that turbot has is composed of 3 genetic clusters, here shown on top. Sole also is composed of 3 genetic clusters, whereas brill only is composed of 1 genetic cluster within the same distribution range. So conclusion from this study is that turbot has 3, sole has 3, and brill has 1 stock, so they differ in stock structure. And so the management strategy should be based on the species with the strongest stock structure, which in this case is turbot. Okay, now over to the overall conclusions of this presentation. So landscape genomics allows to map the distribution of genetic diversity across large and ecologically diverse landscapes. It helps us to predict loss of genetic diversity. It helps to determine the spatial and environmental drivers of adaptive potential. And therefore, it helps to propose management scenarios, such as assisted migration, spatial planning, and in case of plants, I haven't shown any example of plants, but of course, you can also use it on plant species. It can show which genotypes you have to plant, for instance. And therefore, landscape genomics is a useful spatial planning tool to predict and mitigate effects of anthropogenic change on biodiversity. So that was my short presentation. I would like to conclude with some announcements. First of all, if you're interested in landscape genomics or biodiversity conservation, feel free to contact me. It would be nice to discuss any potential collaboration. Second, we also have our seminar series here at New University, and we're looking for speakers. Primarily, we're looking for 1-2 speakers for the next semester. And third, we also have an available PhD position, which you can find under this link. After the talk, I will paste it in the chat. And we primarily encourage female applicants with interest in or experience in computational biology. So we encourage those candidates to apply. And also for speakers, we're looking for a gender balanced panel of speakers, so both male and female scientists. And you can contact me by email. Thanks for your attention. Looking forward to some questions, and just want to show in the end where Noor University is located. It's here at 67 degrees latitude in Norway. And so this time of the year, it's winter and it's still pretty dark outside. So thanks for your attention, and I'm happy to answer questions.
Okay. Thank you very much, Professor Andre, for such a important and relevant talk today on biodiversity loss and climate change interaction, and how adaptive potential and landscape genomics can help us to restore the biodiversity. Thank you very much. Now, we have time for question and answer. And if somebody is interested to ask some questions to Dr. Andre. No? Okay. No questions? Okay, then. Thank you. Thank you very much, Professor Just Andre, once again, for giving this talk to in this conference. Thank you very much.
You're welcome. I'm happy all was clear.
Okay. And maybe now hand over this to the Professor Jonathan Lal.
Yes, sir. Thank you very much, sir. A very well done presentation by Professor Just Andre. Thank you, sir, for your presentation. It was very interesting. Now moving on to the next aspect of this conference is I'd like to introduce and welcome our chief guest, Dr. Sasmit Patraji. He is the member of parliament of the upper house of India. And a few things I'd like to introduce about him is he's also the member of the Parliamentary Departmental Standing Committee on Human Resource Development, which covers the Ministry of Human Resource Development, Women and Child Development, Skill Development, Entrepreneurship, Sports and Youth Affairs. He's also a member of the Parliamentary Joint Committee on Office of Profit. He's also the youngest Rajya Sabha MP member of Parliament from the state of Odisha. So that is a nice record, sir. He's also the national spokesperson and media coordinator of Biju Janata Dal, which is the national party of which he represents. He's also the secretary and member of the State Council of Biju Janata Dal, as well as the member of the BJD's IT Wing Core Committee. And at least from my point of view, one of the most important and prestigious things to say is he has also presided several times as the chairperson of the upper house of the Parliament of India. So that is quite an honor, and it's quite an honor to have him here. He has work experience of over 17 years in academics, business research, industry, and corporate sector. Also with the United Nations Development Programme, Government of Odisha project, apart from political work and social work. He was also the Dean of the Xavier Emblon Business School, the first Indo-French business school in India, a joint collaboration between Xavier University, Bhubaneswar, and Emblon Business School, France. Now, sir, if this conference was offline, I would have handed you some bouquets, flowers. But I can later on send an image file or courier flowers to your kind self. One of the most important things is that biotechnology, bioinformatics, and biochemistry, for me, at least for me as a scientist, is the future of this country. And I think when we talk about the future of this country, it is through politicians who actually take forward our country. So it is very important to have a politician on board in the form of Dr. Patra to join us so we as scientists can interact with him. And it is an honor, sir, to have you as a chief guest. And I hope you enjoyed the previous lecture, and we look forward to hearing from your perspective on science, policy, and where we stand. And with your support, how can we take biotechnology forward in this country and give it more importance like it is in the West, sir. Thank you, sir, very much for coming, sir. Patra, sir, now I hand over the floor to you. We look forward to hearing from you. Thank you very much, sir.
Thank you so much, Professor Dr. Jonathan Lal. It's a wonderful opportunity, and I believe the words that you've already spoken are so beautiful that the flowers at times would fade before the words that you've used, which I'm totally unworthy for that, because I've always considered Schwartz my family. And I started my career with Schwartz, which was Allahabad Agriculture Institute deemed university in those days. I see Professor Lawrence and Professor Kapil, and I remember Professor Ramtekar is there. So, and Dr. Tripathi, and you all family. So it's not about being a chief guest. I believe I've had the honor and the privilege, and thank you so much for inviting me at this wonderful opportunity and giving me this wonderful opportunity today. Having been an academician and haven't had a lot of science and not as bestowed richly as all of you people are in science. So I'll not try to hazard a guess as far as the science is concerned of biotechnology, biosciences, or for that matter, even bioinformatics and biochemistry. I learned none. But what I'll do is I will bring it from the perspective of policy, of governance, where I see as a parliamentarian the field of biotechnology, bioinformatics, and biochemistry going forward. And I would like to, because of a paucity of time, I would like to focus not across the spectrum. It's a very wide spectrum, but probably focus firstly on biotechnology and then a bit of bioinformatics. Now, uniquely, Professor Lal, how much of time would I have? I mean, always give time to a politician. Never allow him free.
You have 25 minutes, sir, but you're the boss.
Right. I really appreciate that. But just buzz me if I just forget the time, because unfortunately in the class we have, but you're the monitor. I'll leave it to you. So as far as biotechnology, or today we talk about these fields of biotechnology is concerned, I think COVID, despite the fact that we have always talked about COVID being an enabler or an opportunity in disguise, one fact that actually remains is that if today India has a very quick turnaround time in terms of COVID management, and I'm not focusing it primarily from the biotechnology perspective, but from a generic perspective, if we were to look at it, it is primarily to do with the kind of work and the scientific temper and the scientific work that India has done as compared to the resources we have. So just like Schwartz has got very limited resources that it can really put into research and development and things like that. But despite that, when you look at the return on investment or the return on the capital employed, it's huge. So that is why today I believe COVID presents a unique opportunity for India to leapfrog many nations And when I say many nations, I would rather focus on US and Europe in terms of finding out new innovations in the area of biotechnology. Now, let's say, for example, let's go back to the basic concept of pharmacy and then look at it from the biotechnology perspective. Today, the world over, whenever it came to issue of COVID and there was a short supply of pharmaceuticals, then everybody turned back to India. From Trump to the European past, everybody called up that, India is our best friend and we need to get doses. And whether it's the Serum Institute, whether it's Mr. Poonawalla, whether it's others, everybody pitched it. And we also exported millions of kits, millions of doses of medicines abroad.
I think we have lost Dr. Patra. Let me just check. Yes, sir. Yes, you're back on, sir. Sorry.
Yeah, I don't know how. Yeah, we just lost the whole thing. So I was just talking about, I'm sorry for this interruption. I'm very sorry. I don't know what happened, Mike. The whole thing just stopped for a minute. So I was talking about something that I was looking at in terms of India becoming a biotech hub. And I would like to refer to that because that also gives us a glimpse into the policies and the governance mechanism and the way India, and I will talk from the perspective of India primarily, that it's looking at in terms of making India as a biotech hub. So tomorrow, whatever advances and whatever developments that come about in the biotech sphere in India is also going to have a reflection from this article by Dr. Renu Swaroopji, who is the Secretary of the Department of Biotechnology. And one interesting factor she's talking about is the making of a biotech hub of India. Now, where does that come in? That comes in from evolving as a model of pharmacy for the world to becoming the center of cutting-edge research in biotechnology. So for long, we have always looked at pharmacy, but we have never been able to extend it into the biotechnology sphere, where we actually invest more and more in terms of resources on research and development. Now, let's say, for example, if you were to look at it in terms of the COVID pandemic itself, I was just referring to how the researchers and innovators brought about a lot of low-cost diagnostic testing kits, manufacturing of PPE kits, therapeutics, the clinical trials, the kind of serums that were actually developed both by Pune's Serum Institute, as well as by Bharat Biotech Covaxin, as well as by Zydus Cadilla's Zycov-D. So all of this rapid, quick movements by the pharmacy / biotech concerns, if I were to say, has got a lot to do with the kind of focus that today India is trying to make vis-a-vis moving and transitioning from being a pharmacy-centered approach to becoming a more biotech-centered approach. Because for long we felt that if we just built, because what used to happen is, whether it was drug discoveries, whether it was in terms of development of therapeutics, we used to borrow the patent. We used to either do that, or we would probably commercialize it, or we would get it licensed. But now you find Indian firms in the area of biotechnology taking that step forward, the risk of staying invested on a product or development of a product, and then seeing it through the face of the intellectual property rights, and then after going into mass production. So therefore, if you were to look at it, so that's one area where I believe biotechnology has a lot to contribute in India. And going forward, I believe there is this innovation ecosystem that is slowly being developed in India, encouraging young scientists to actually be willing to fail. In India, the problem is we have a fear of failing. If a young scientist comes to Professor Lal and says, you know, you give me an equipment, I'll probably invest 50 lakhs on that equipment and its ancillaries, but I'm not sure whether what I'm working on will actually come about or not. So Dr. Lal will also have a difficulty explaining to the finance controller and to the vice chancellor why he's spending 50 lakhs when there is no certainty of a product coming out. It may not, but that's science. And I think India is coming to grip with that kind of an ecosystem that we are ready to fail, provided in the long run, we stay invested in bringing about the changes or the research and development in the area of biotechnology. Because I had a little bit of a stint with a biotech concern way back in 2006. And 2006 was the time when people used to think about biotechnology. I believe Professor Lal at that time was the student in biotechnology. And everybody said biotechnology is the next future. We're talking about biodiesel, talking about nutraceuticals, we're talking about curcumin and catechins. So we're talking about all of that. And I remember when I went to work for a corporate entity in Calcutta, and this entity was working in terms of developing agricultural products, nutraceuticals primarily, and then using them in terms of wellness hygiene programs. Now, one of the areas that really came about there was that, you know, everybody wanted to become Kiran Shah Mazumdar overnight. I remember at that point of time, Syngene and others, or even Aldrich and others were all, you know, I mean, everybody was collaborating with them, thinking that, you know, they have the, what you can say, the holy grail to the next level immediately. But the question is, we have to understand, and I'm sure over the last 14, 15 years, we have realized that biotechnology, or for that matter, bioinformatics, is on a very different plane than a quick fix method. And that is where I believe the policies of our country has slowly started accepting it. I'll take you back into 2002, 2004, when Jatropha as a seed was being considered as the biodiesel formula for actually, you know, bringing about the biotechnology's answer for motoring the locomotives of our country. And everybody said, oh, all this diesel is out and electricity will not be required. We will blend biodiesel, and with biodiesel, everything will change. And biotechnology, you know, would turn it around. But as we move forward, we realize that, look, biotechnology has some answers, but it's not the answer. So therefore, going forward, the policies of the government, as I see it taking place, is looking at those specific niche areas where they feel biotechnology can actually be the answer to many of the issues. One, of course, we have talked about COVID. There's another aspect I want to say, which is regarding the Biotechnology Industry Research Assistance Cell, which was established under the Department of Biotechnology in 2012. Now, BIRAC, as it sees, is actually bringing innovators and funders on a common table. Now, when you have funding agencies and innovators coming together, and a corporate entity, when it's coming for kind of a product, it's coming in for some kind of a technology, and the government is trying to enable it, I think we are missing the mark in terms of the policy there. Like, for example, if you want the government to play matchmaker on intellectual property rights, I think that's not the right strategy. I would suggest that probably there has to be a dedicated think tank run by professionals, not by bureaucrats, run by people who understand biotechnology. For example, I said as a disclaimer right away, I've never studied biotechnology, so whatever I talk about it would be from the policy perspective. But many times when we have bureaucrats running organizations, which are scientific organizations, technological organizations, but they do not have they haven't learned ABC of that technology, so they try to administer a technological or a research and development. That is where we have a problem in our country. I believe, therefore, the policies have to look at, let's say, for example, we have this Development Assistance Council. Now, that council, again, has to be let go and allowed to be run by people who understand biotechnology, bioinformatics, and biochemistry, and give them the free hand. The problem is here we try to micromanage everything. The minister who has no idea about biotechnology will talk on biotechnology. bureaucrat who is 3 months in the Department of Biotechnology, then 3 months in shipping and 3 months in coal and mines and 3 years in something, that person will also claim ownership of biotechnology. I believe, therefore, the policies and the advances in India that can actually be made is getting stunted. It's because the brains who should be there engaging. When I talked about this assistance council, it should be the innovators, the young scientists who come up with models, who come up with prototypes, and they, when they come across with the angel investors, like it happens in the US, if you were to go to a place like Stanford, on one side are the venture capitalists, and on the other side is the university. So Professor Lal gets up and then he goes across to the venture capitalists, the angel investors, sits down with them, agrees on a certain agreement, and the university has an incubator which helps incubate the facility. So you're looking at an ecosystem where we are developing talent, nurturing it, because let's say, for example, somebody may be a very good innovator in terms of biotechnology. The problem is that person aware about the intellectual property rights? Maybe. Is that person aware that once the person has patented it and it enters into an agreement with a large corporate entity, in the fine print of those 500 pages, what is exactly written and where that person will get caught, if not today, after 5 years, and suddenly one fine day find that instead of a 90% stake, the person is left with a 5% stake. So these are the nitty-gritties of which we have to create an ecosystem that will take biotechnology and biosciences forward. One more approach that I believe is very important. Now I want to come about bioinformatics. I won't take a lot of time in terms of dealing with it because I'll just focus on the key areas and the nuts and bolts of it. In terms of bioinformatics, the question is right now, we even now look at bioinformatics as a standalone domain. I believe there's so much of synergy that bioinformatics, artificial intelligence, and big data can actually integrate. Somewhere down the line, the data, and I'm privy to some discussions that happens on the data protection law that is going to come back about in the country. And the data protection law is very, I mean, it could, if it's put in the way it is actually coming in, it'll be a death knell for the bioinformatics. Because let's say Professor Lal has the data in his database on anything. Let's say the student database is with there. Now, tomorrow, whether Dr. Lal can actually transfer that data to someone else, if it is that data is with him, is the ownership with him or with the students? And if he transfers it to a third party, then what happens? So if that is the basic construct of data protection or data ownership, and we were talking about bioinformatics today as the next level of, because bioinformatics is nothing new. We were talking about it, and basically we have been talking about how bioinformatics can be made more and more intelligible. The question is, when we look at bioinformatics, has enough been done in this country? I do not think so. And the simple reason is we look at bioinformatics as a standalone. I would suggest today, when we have information technology, why can't we embed information technology and bioinformatics more integrated manner? When I say that, what do I mean? Artificial intelligence today applications are focused more on products. Whether that is products or online platforms, we have artificial intelligence coming in. But how do you really wed artificial intelligence, big data, and bioinformatics is a challenge that is going to be there in the coming days. For example, I'll give a very small matter. Over the last 3 years, there are more than 1,280 publications, 56 patents. There are major programs like the Indian Biological Data Center, Biotechnology Information System Network, the MANAV, which is the Human Atlas Initiative, the Indo-Japan Data Center Program, the DBT APIX BTCIC, and the Northwestern Himalayan Bioinformatics Grid. Everybody is working in their own specific areas, and I do not want to go into the specifics and the nut bolts of it. But are they talking to each other? Are they engaging with each other? Is there data sharing? Are we able to map our own areas of competence and therefore share data? So I believe somewhere down the line, it is going to be very critical if you want to really develop bioinformatics the way it is. Various departments, and I'm looking at it from a policy perspective. I'm not looking at it from the science perspective because I do not understand the science. But I look at it from the policy perspective that if there are 6 or 7 domains in bioinformatics work being critically done by the government of India, why is there no integration? Where are the universities and these data centers? Where is the bioinformatics work and its engagement with the academics, engagement with the incubators, engagement with the potential investors, engagement with the potential developers, engagement with the potential entrepreneurs and innovators? So the hub and spoke model of the government facilitating it, and even now if you look at bioinformatics, most of the data information that is there, it is government controlled and government owned. If you're not willing to give out the data, for example, let's say someone wants to really work on a specific area of bioinformatics, and that information is guarded and solely under the custody of a city, a corporation, or that of a state, or that of the central government. And there is a developer who says, look, if you were to give me that data, I can work on that and I can give you back some kind of a solution. The decision sciences we can use, or we can use artificial intelligence. For that, you do not have a scope. So it is always like, it's my data, I'll protect it like a holy grail. It's your data, or you have to give it to me. So no one wants to share, which I usually say that how many files do we upload on the internet and how many files do we download from the internet? That ratio will tell you how much we are contributing to intellectual capital gain and how much we are taking from the intellectual capital that is already existing. And the third area, and with this I'll close, I believe the universities at the educational level have been doing substantially enough in terms of helping the students, in terms of encouraging talent and making them aspirational. But that is where the government of India, and I understand there are always challenges for universities when they go looking for grants. Now, if you want to, so the government cannot fly airplanes, the government cannot run hotels, and the government cannot do the biotechnology itself. I've always believed the government should be a facilitating agency, but the funding and the resources it should provide is to platforms like universities, incubators, and other think tanks where this kind of a work can be actually taken forward by the experts. The government role, unfortunately, is to control resources, to micromanage resources, and also to determine whether Dr. Lal and others are competent enough to use those resources. So if I'm not aware about biotechnology, how do I determine whether the Department of Biotechnology in Schwartz is competent enough or not? So I believe we have tried to be the master of law, now jack of all trades, master of none. That has been the policy. So I believe the policies, at least in the scientific space, have to change. I'll just digress a bit. And I want, because I'm also, a couple of days, we'll be doing a standing committee on the Tokyo Olympics, and I happen to be part of that committee as well. And usually I find that when I talk to federations, most of the federations are run by either businessmen, corporate heads, or probably by politicians. The bigger question I always ask as a member of that committee is that, look, you have no idea what is a volleyball. You've never played volleyball, but you're the president of the Volleyball Association. You've never played a badminton, but you're the president of the Badminton Association. You never played soccer beyond your school, but you're the All India Football Federation chairman. The problem is, anywhere you find there is a competency required, I'm not saying it should not be, but be there where you can facilitate. Do not become a line manager. If I were to use my business management terminology, be a staff manager, not a line manager. Go into staff operation, not a line operation. Line operation of biotechnology belongs to Professor Rubina Lawrence, belongs to Professor Dr. Kapil, belongs to Dr. Koshi or Dr. Ram Teke or Dr. Tripathi. If I, as a management professional, try to appropriate it, I'll make a very poor show of it, and they'll be also pulling their hair ki yaar ye kahan ja raha. So I think as a policy, we should be there to facilitate experts like you, take in the data, create an ecosystem, provide the relevant requirements. Let's say the department develops something, it says, how do I really, I have to file 5 patents. It is an international patent. There's a lot of cost. Can the government come in and help me? Now, you cannot write mails and you cannot call people and after 3 months you'll get a reply. No, it has to be 24/7. There has to be a timeline. There has to be a charter. And I believe if the advances of biotechnology riding on COVID, post-COVID, also in the areas of agriculture and other areas are to come about, it has to be done with a very concerted effort where the bureaucracy, the political classes take a step back, allow the professionals, the people who understand biotechnology to take up the reins of the operations and just facilitate and create an ecosystem. Otherwise, we will again miss the bus. I believe right now the time is ripe that post-COVID, you have a situation where we can actually leverage our position from being a pharmacy-centered nation into a biotechnology, biotech-enabled nation. We should not be there to just be a copycat and take their formulations and run it. We should be able to develop our formulations like we have got some people doing it and become world leaders as well. So with these words, I want to thank you for giving me the time and opportunity. And wherever and whenever I can be of any help, please let me know. My deep regards to Dr. Lal and to the entire team at Schwartz and to all the members and delegates of this international conference. Thank you.
Thank you, sir, very much for your very enlightening talk. And I think most of the audience members, in fact, all of the audience members now realize why we need a politician in such scientific conferences. I must appreciate the knowledge level you have shown and the understanding of us as scientists, where the frustrations we have, where the bottlenecks are. There are two questions from the audience, sir. So I'll read the first one. It's an anonymous attendee. It says, sir, major entrance exam in the country for bioinformatics are based on life science only, not giving enough emphasis on bioinformatics. So how can we, the bioinformatician, work in sync, as most of us get filtered out in the selection process in public and private domain institutions? This is indeed a problem, sir. Over to you, sir.
May I suggest that this being a very critical matter, may I request either if through the department, or this information can be shared with me, I will take it up with the concerned Minister of Science and Technology, Dr. Harshvardhan ji, and the concerned person can also be in touch with me. And I'll probably do whatever we can in terms of escalating it. I do not know whether we can resolve it right away, but at least raising awareness about that and trying to pitch and push with the ministry and the department. At least we'll be able to get the answers, A, and B, if the answers are not satisfying, at least push them to allow this kind of a request to be undertaken, because this is not an individual request, it's a generic request which must be impacting hundreds and thousands of professionals and making them devoid of the opportunity that they should richly deserve. So I believe, please share with me these details, Dr. Lal, through your office, and I'll be very happy to take that up.
Thank you, sir. Anonymous attendee, please send the conference organizers email, and sir will try to resolve this as much as possible. The second question, sir, for you is from Mr. Dheeraj Sinha. Thank you for such a nice lecture. My question is, where we lost the way in science with respect to other European countries, why are policymakers not able to learn in the last 70 years, and we are still struggling for basic infrastructure? Sir, over to you.
Two reasons. One, the policy was always myopic. When we started out as a nation which used to have very low resources. And when I say low resources, I'm sorry, my electricity ran out. So
low resources, sir.
Yeah, low resources. But I think you can still see me. Yeah.
Yes,
so, yeah, let me just pull back my screen. So, the fact is that when we started out as a nation, we had very few resources there. And the primary area at that point of time was survival, more than probably anything else. When we talk about survival, we talk about food security. I'll probably take a bit more time in terms of analyzing it, because this has a wider reference, because you come up a lot of students and professionals saying, why didn't we ride the technology bus? Why did we got derailed? I'll tell you something. Kalahandi, in the state of Odisha, in the 1980s was known for starvation deaths. Not very long ago, in the 1980s. Today, it is the rice bowl of Odisha. You know how this transformation came about? Because we stayed and kept invested there. Similarly, if we were to look at it, in parts of Odisha, there were no railways, even after 70 years. So now you've got districts in Odisha where you have the railways being laid down for the first time since Indian independence. So therefore, as a nation where resources were always scarce and still are, we always used to focus on the social welfare, probably more than the resources in the research and development sector. Secondly is, whenever there is, like, for example, now there is this epidemic and crisis, so most of the resources get slashed out from the research and development fund, because that's the soft target. Now, by saying all this, am I trying to justify that, no, we should not invest and we should not stay invested? No, Because if we did not have the private sector, am I audible?
Yes, sir, you're audible, sir.
Yeah, so if we did not have the private sector, then the government sector would not have been able to come out with these kind of innovations and solutions. So therefore, I believe the government policies need to be more focused on investing in technology and in research and development. Having said that, I would also, probably give the benefit of the doubt also to the governance and the policy makers, where till probably the 2000, at least till the 1990s, when we had the economic reform, till such time we used to pledge even the gold of our country in order to make the balance of payments. If you remember the time when Professor Madhu Dandavate was the finance minister, India had to pledge its gold in terms of honoring its balanced state of payments. So that was the situation at which we used to work. Having said that, now is the time, over the last 15, 20 years, that you see there has been remarkable change in terms of resources and funding. And the second aspect that we would suggest is in terms of talent. How many, Professor Lal, you tell me, how many people or how many parents ask their children even today to stay invested till 26 years, get a PhD in biotechnology and stay invested for another 10 years doing research? Today, the sociological fabric is such that the point the girl or the boy becomes 16, they say, wait, doctor ban ja, engineer ban ja. Doctor engineer nahi to MBA ban ja. So the problem is the mindset of getting the right talent is also necessary. Today, you have the best talent with 90-95% in scoring going into engineering and clicking computers. I have no problem with that. They are perfectly doing well and we're doing a good job. But you have the best talent going into medicine and functional areas of work. Where does the best talent really come back into science? And science, to be very honest, people who are invested in science, it's a huge sacrifice. People, I have seen people, I mean, I'll give a very small example. Dr. APJ Abdul Kalam. The man never married. The man lived on only 2 pairs of uniforms all his life and became the missile man. He invested everything so that he could give it to science. So science requires sacrifice. And I believe apart from only resources, there's also a mindset and a talent. Right now, do people have the right talent? Yes, we have. Do they have the mindset to stay invested for long? I doubt. I think that is where institutions have to really make students become more aspirational and say, look, science is not a 2020 game that one day you come in and within a short period of 4 or 5 years, you'll become a successful scientist. To actually bring about a good paper, a scientist spends more than 10 years. So the question therefore is, can people stay invested in science and thereafter bear its fruits? I think it is more about the quality than the quantity. So it goes both ways. I believe policies need to be framed better. Resources need to be invested more. The ecosystem needs to be improved. And the kind of talent needs to be further harnessed, honed, nurtured, that they stay invested in science.
Thank you very much, sir. I totally agree with you. When I was in Europe some years ago, I was on the bus. And then the bus driver told me, he asked me where I am from. I think that was back in 2005 when I first went to Europe. Then he asked me, where are you from? I said, I'm from India. He said, oh, yes, I go there on vacation every other year. And he's a bus driver, right? In India, a bus driver cannot afford that. So there is indeed a lack of social security in this country, because I've seen students in Europe, they drop out of after high school, they just do a diploma in carpentry or something. And they are well paid enough that they have a sense of life security. And therefore, those people who actually enter the university are only those who are brilliant minds, those who actually want to do research, who are interested, not because of peer pressure. In India, we will have to change the whole social security system on how we look, because everybody, in admissions, we also have admissions here. People come and say to us, shall I take admission in this program? Does it have good job prospects? So they don't really care whether they're interested in the program or not. They're more interested in which degree program would land them a job. So, and then there is the other side of the category, those who are not able to find jobs, they start doing PhDs. So this aspect also has to change eventually. And I think that can only change once we have a good economic development. Now, when we talk about Israel, sir, Israel, they really focus their GDP on R&D. And since you as a politician here, one of my requests on behalf of the scientific community is if you can proposed in the parliament as per your convenience that if they could increase the GDP for R&D rather than taking away from it. Because if they invest more in R&D, we would have good research and more funding for capable institutions. Another aspect, sir, which I'd like to request is the general discriminations between private, central, and state universities. The government prefers to give more to the national institutes of importance, the IITs, IIMs, and others, and perhaps state and central universities, but sometimes try to, of course, private universities do get funding, but not compared to central institutions, among other. Rather than establishing new universities, we should strengthen existing infrastructure of universities we have. So that is another request I'd like to say. Another problem which I have been facing coming back from Europe, this is me talking not on behalf of Schwartz, is when I worked in Netherlands for 10.5 years, coming back over there, we had academic freedom. Universities determine the quality and the strength of programs we develop. Accreditation for us is more of an optional aspect, whether we want to go for it or not, which of course we do go for it. However, what we're seeing the government's doing, this is my personal view, is that they're linking their accreditation with funding. That is, if universities do not go for accreditation, their funding gets reduced or stops. So that basically, in essence, makes us universities more or less into a degree college, and which I do not think is right, because then you also restrict our academic freedom on the nomenclature of programs, the programs you want to build, what the infrastructure we want to strengthen is all dependent on government bureaucracy. So that somehow eventually has to reduce, because India has a huge potential, huge talent pool, but we're not able to tap into it. One of the reasons is there are talent who are very good, but they have financial restrictions due to which they're never able to explore the talent in the scientific community. So there is a sense of what we lose there. For example, when I was working, another aspect is when I was working in Netherlands in cancer research, we worked with doctors. Of course, we were doing molecular diagnostics, DNA damage, and we're looking into all those things. But the doctor never dictated how we're supposed to do our research, right? When we come here, when we want to work on, human models and stuff, of course, doctors are there, but the MCI is put as such that as if they are overlords rather than our collaborators, they become overlords. Because in science, there should not be any boss. There should be people who collaborate with each other, find solutions to things. And I think I totally agree with your lecture at the very start, that bioinformatics was in silos, it was in different places, but now the advent of systems biology, we're involving biophysics, mathematics, biology and stuff. So a lot of stuff is coming in and being integrated. In the start, in the early 2000s, everything was treated differently, but slowly we're getting there. We're becoming interdisciplinary. We're mixing all things together. Another point, sir, I wanted to request is, the risk-taking point. We know that startups, of course, the government, and they're all working on Startup India, Atal Innovation Scheme, among other stuff. But the risk-taking is very risky in the Indian scenario, because you don't have social security. So those entrepreneurs who want to take risks, they go get incubated. Especially biotechnology is a product development-oriented field, whether molecular diagnostics and human research, animal research, industrial research, biofuels and stuff. They're all industry-oriented, all product-oriented. So if any students want to start up their own companies, start businesses, they're really fearful of the risk that if, as you said, that, if they invest 50 lakhs, and will I give it to them or not? So, and that is one of the major reasons that the really brilliant ideas stay back, because the students do not want to take risk. If government can strengthen further those schemes so that the students can become better risk takers, so that once they take a risk, they're not jobless, unemployed if they fail. They get another opportunity. If something could be looked into that, sir. And another aspect, sir, in Europe, I noticed is the privacy issues. They had very well strengthened privacy norms when it comes to that. And since we're going to work, I work with patient data. We work with patient privacy is utmost important, but it is not well executed. And that should be better regularized, I think, sir. That I just bring these few points because I know you're super intelligent and you present a lot of important issues in the parliament. And I said, instead of asking questions, I like to just let you know my view of things we would love to include. And sir, the feeling we scientists sometimes get is that when we work with the government, when we apply for grants, for example, I also have grants from DBT among other agencies, you start feeling that they are your bosses rather than they are the investors, the funding agency. Because, for example, when I worked in Europe, Framework Program 7 or Horizon 2020 grants we got, it was pretty straightforward. It was, there was no bureaucracy. They were pretty targeted, focused, and they were pretty humble, and they made you really comfortable in the research. They only focused on the quality of that. That's about it. But the feeling we get here is, at least what I get here, is that then you feel like you're talking to your boss, which in fact they are not. They are a funding agency. And a trivia thing I'll tell you, sir. As you said that the bureaucrats hold positions in different organizations without having expertise in that. Funny thing happened several years ago with our Honorable Vice Chancellor. He found out he was a member on the BCCI, Board of Cricket Control of India, to select cricketers for a few terms, but he had no idea why he was put there. So he sent from his network people who were actually interested or had some history in cricket, and he never went to those meetings because he said, I'm not the expert in that. And I think more of our officials should look into, when they get into boards and committees, how much their expertise or administrative value will really work there for the development. And I think if these things, among others, sir, as you have mentioned, will be addressed, India indeed will go way ahead in terms of R&D, sir. Thank you, sir, very much for your time. I just wanted to conclude, sir. We really appreciate, and sir, you have been a very humble chief guest for us, and you, sir, have been indeed a Schwartz family member, and we do really appreciate all the support you have given us throughout the time. And we look forward and congratulate you on your political career. And sir, we will fully support you. And we look forward to see you very long in the parliament so that you can help us academicians more and more. Thank you very much, sir. Thank you for your time.
Thank you very much, Dr. Lal. And let's engage, let's work together, because I'm putting together a platform in Delhi called as the Parliamentarians Group on Technology. Yes, so I do not want to probably keep it as biotechnology or IT. It's just technology. And I will invite you and your team, once we are in the formative stages, to actually engage and use the parliamentarians, their services, in terms of emphasizing the policy advocacy. What we have specified is the need for policy advocacy and being able to influence policy that becomes more user-friendly, so to say, so that intellectuals, think tanks, researchers, scientists, academic institutions should not feel that they have bosses sitting in large bureaucratic concerns, but rather it is an ecosystem where everyone takes out something with a win-win situation for the larger interest of humanity. Because ultimately, that's what we are all for. That's what science is for. And I completely believe that we need to work together. I'm open to working together and working on this, taking this forward. So God willing, 2021, we will all work together more closely, and we will provide the opportunity for you and your team to also present your ideas to the fellow parliamentarians at a place we'll put up together where you can engage and turn this into a policy advocacy for the larger interest of the biotechnology field. So thank you so much for your time. Thank you everyone at Schwartz. It was a pleasure and a privilege being here. Thank you. Namaskar.
Thank you very much.
Thank you, sir.
I now hand over the session back to Professor Ramateke, sir. Back to you, sir.
Thank you very much, Professor Lal. Am I audible? Am I audible?
Yes, sir. Yes, sir. You're audible.
Can you hear me?
Yes, okay, sir. We can hear you.
All right. Thank you. Now our today's session last speaker is Dr. Arnab Bhattacharjee. And before I invite him, I would like to introduce him to you. Dr. Arnab Bhattacharjee is an associate professor at School of Computational and Integrative Sciences, Jawaharlal Nehru University, New Delhi, India. Before joining JNU, he served as assistant professor as a DST inspired faculty at IIT Delhi. Dr. Arnab earned his PhD from University of Delhi in 2010. He did his postdoctoral study at Weizmann Institute of Science, Israel, and Theoretical Physics Division in Sweden. His lab focuses on transport and trafficking phenomena in biological systems through the study of structure, function, dynamic relationship of complex biological systems that have long-term implications for combining medical conditions such as Creutzfeldt-Jakob, that's mad cow disease, Alzheimer, and cancer by using computational molecular biophysics techniques principles. He has received many accolades and awards like incentive awards for publication in 2017 by Department of Biotechnology, Indo-US Science and Technology Forum Award for supporting Indo-US Symposium on multi-scale simulation and mathematical modeling of complex biological system in January 2019. Oh, he has a very good work. And now I invite Dr. Arnav Bhattacharjee to present his talk.
Thank you, Professor. I hope I'm audible. Hello.
Yes, you are audible. Yes, you are audible.
Screen is visible?
Yes, it is.
Okay, so thank you once again, and just sorry, let me minimize my photo. Great. So thank you once again, and I actually enjoyed being in Shorts last year. Dr. Tripathi, who is a friend of mine, invited me last year, and unfortunately, because of COVID, we cannot essence of our work that we are carrying out. So basically, okay. Thing first is that I apologize if any internet connection is problem is there, because I'm actually not in my home institution, and actually I'm traveling. So there could be small problems, but let's see. So basically the idea is that after this long COVID time, I was trying to get out of JNU and have a journey, have a trip. So when, and probably many of us are actually bored, and they are looking for escape opportunities. And when we plan something like that, we typically plan for our destination, and we really focus on the destination and how it should be. We completely concentrate on that place. And during the course, we often forget the importance of the road to the destination. So in my talk in next 20-25 minutes, I would try to emphasize that the road to the destination is equally important. So this
Oh, we lost you, Dr. Arnab.
So this kind of interaction, yeah, is audible?
Yes, now it is.
Right. So the destination is, as I said, the specific protein DNA complex, which is important and essential for numerous different DNA metabolic processes, kind of transcription, DNA damage repair, replication, gene editing, and many more. So the point is that obviously this destination or the final product, as I say, the journey is often more beautiful than the destination. I'll try to show you that how the protein will try to find their destination, or when I say the destination, that means the specific target site or the specific DNA sequence out of the whole genome, which is extremely long, extremely crowded. And how do proteins find their destination And I'll try to emphasize the different types of the modes of transportation and all. So this is the DNA system that we have in our cellular condition. The chromatin is extremely long, extremely complicated, compact structure, which has lots of the basic unit is nucleosome, which are basically 8 histone proteins, and DNA is wrapped around that protein. So right. So this is extremely super complex structure, and there is not much space that the protein can, apparently it seems it's very difficult to move into in this situation. So the question is, how do proteins find their target site, right? So the question can be answered or can be understood in this way, that when you try to move from one place to another, we have let's say these days Google Map, which is extremely convenient. And if you see, for example, this is a Google Map from New Delhi to New Delhi railway station to Jawaharlal Nehru University. And if you try to see it, there are multiple ways to reach JNU from the station. And not only multiple routes, there are multiple ways to commute, right? So you can hire a cab, you can take when you that means you have different ways to do this. Now, obviously, if you walk all through the way, so you will reach. It will take a longer time, so the time will be higher. But on the other hand, you will actually experience the road. You can feel the road. You can feel the journey. So obviously, that means that this walking is actually, although this is the slowest process, but you can enjoy the journey, or you can. So similar thing happens in case of protein. So when protein moves one-dimensionally, or it walks kind of on DNA, we call it it's a sliding dynamics. So during this sliding dynamics, what happens is protein moves along the DNA, right? So when it moves, it basically moves along the DNA, and it also advances along the DNA length. So that means, and basically because it moves one-dimensionally along the DNA length, it is extremely slow process, but the protein actually gets a huge chance to actually see all the DNA bases very carefully. And then this is extremely beneficial for the protein to find out each and every its target site, because it can scan each and every basis very minutely. So this kind of dynamics gives you this kind of pathway where you have a spiral motion along the DNA, and depending on various factors, the geometry and the length of this path changes. So experimentally also, this kind of motion has been found out, and this paper was published in Nature Structural Biology by a group of Sunny Z, but they have shown that this protein actually, they agrees with that, the factor that the protein actually rotates around the DNA like this during its diffusion. So also another fantastic way is that protein, like us, it takes shortcuts, right? So when I say shortcut, it means that the protein has to scan let's say the left DNA, but then this DNA, let's say, is extremely long, and so the protein can actually bypass if another DNA segment eventually comes close to this segment, and it forms a kind of bridge complex, and then transfers to another DNA segment, and thereby it can basically bypass scanning huge length of the DNA. So it takes kind of shortcut to make its way to its target site. So we call this kind of motion as intersegmental transfer. We have also looked very minutely to the traffic situation. That means when you see you have a protein, you have a DNA, and then the protein moves along the DNA. So there are many more proteins which are there, which can interact with the protein, with this protein, and it can changes, or it can affect the diffusion of the protein along the DNA. So we have looked into many possibilities, and what we found is very interesting is that, like, if you try to see, if you are in a on a way where the route is extremely crowded, it takes typically very longer time to reach your destination. In contrast, if you have a very extremely crowded condition in the cell, then this kind of situation happens because of the entropic forces, the crowders and the DNA. In between that, there is a depletion layer which forms, and this within the crowder molecules or the other proteins, they are not allowed to enter into this depleted region. So what we found that the searching protein, when it enters into this depleted volume, it moves very smoothly, because the micro viscosity within this depleted volume is completely different and much lower than what you have in the bulk. But in the bulk, you have a higher viscosity because of the presence of this crowder molecule. So basically, due to these crowder molecules, this protein comes, when it non-specifically associates with the DNA, it gets a room which is kind of very empty, although it has a limited area, but it can go smoothly along the DNA surface, which it couldn't be possible if the protein diffuses in the bulk solution. So this kind of, we looked into many other properties. For example, what would be the impact of the physiology of the crowder molecules if the size, shape, and the geometry of the crowder molecule changes, how it impacts the diffusion process, and let's say if the crowder interacts with the protein, what happens, and many more. And so these are some of the papers related to that. So the another possibility is that, like in our real life cases, we have signals in nucleosome. In our genomic DNA, you can have also experience similar kind of situation. So for example, in this case, let's say this red star is the target site, where this green circle is basically represents a protein that needs to find this red star. So what happens is that when I say this red star, it means basically a patch of DNA sequence, a particular DNA sequence, very specific one. And but in the genome, there are many such sequences which are very close to this particular sequence. So those I represent with these orange stars. They are kind of pseudo target sites. So now what happens is that when the protein goes by or close nearby these pseudo target sequences, it kind of assumes, okay, probably this is the target site, and it tries to bind with it. And obviously it looks into it very carefully, and during the process, some time is wasted, right? But on the other hand, when the time is wasted, it finds out, okay, this is probably not the right site, so I have to go out of it. So what happens? That means when you have a sequence that can have some sort of affinity with the protein, then the protein actually stays there for some time. So that means the affinity actually slows down the speed of the protein to reach to the target site, right? So this is in this case, the affinity actually works like a traffic signal. And then there is a famous paradox which says, if you have to speed up, you have to compromise with your stability. So that if you would like to reach faster, you should not have many such pseudosites, or the affinity of the protein towards DNA should not be very high, right? So there should be a balance between these two, which is necessary in a speed stability paradox, and whether this is really true or not. So what we get is that we kind of assumed a system of proteins searching a nucleosome. So in the left-hand side, you can see that this the barrel-shaped structure is basically represents a histone octamer, and on the top you have a DNA segment. So a red pioneer protein would like to find out this target site. So pioneer, when I say pioneer, it means that there are many different transcription factors, but among those transcription factors, there is a class of transcription factor known as pioneer transcription factor, which are enabled to bind with the nucleosomal DNA when the nucleosomal DNA is in a closed state. So basically the DNA wraps around the histone octamer like this, and even in this situation, so basically you can imagine when the DNA is completely wrapped with the histone, so basically the DNA sites are not empty, right? So the DNA sites are already occupied. So if the DNA sites are occupied, how do proteins come and bind? But these pioneer factors have some sort of affinity towards DNA that it kind of come, and even when it is closely bound with the histone octamer, it can non-specifically bind with the DNA and can stay there, which is unlike to other transcription factors. So basically these pioneer factors are known to access DNA even when it is completely wrapped. So we kind of model this kind of system. We have different results of kinetic results of different transcription factors, among which we have 2 pioneer transcription factor and 1 non-pioneer transcription factor. And we estimated that given a normal situation, when you have a DNA wrap unwrap kinetics is there, that means when the DNA can unwrap because of thermal motion, can unwrap from the histone partly and can rewrap, what happens and how long it takes a pioneer factor to find out its target site on a nucleosomal DNA. So when I say this wrap unwrap kinetics, recent cryo-EM study has actually identified this kind of motion, and they have deposited the corresponding structure of the nucleosome in its wrap and unwrap structure. So this is the time that we have estimated. So the lower panel shows you the time on a linear DNA. That means there is no histone octamer. So on DNA, it's a completely naked DNA. That means all the sites are completely accessible. So if you see REB1, CBF1, the red and the green, they represent the pioneer transcription factor, whereas the LEXA, the blue one, represent the non-pioneer transcription factor. So on a linear DNA, when all the sites are completely accessible, you can see that the linear DNA, on linear DNA, the non-pioneer transcription factor reaches its target site much faster compared to the pioneer transcription factor. But the vice versa is not true. I mean, on the nucleosomal DNA, if you see, the pioneer factors are much faster, way faster, or orders of magnitude faster compared to the non-pioneer factor. Now, if you really look into this table back, if you can find that if you see the K of nucleosomal value and K on nucleosomal value, you can find out the affinity of that means REB1 and CBF1 much higher compared to non-pioneer factors. And this result, if you couple it with that observation, then you can say that, okay, probably the affinity towards DNA is not really bad. And on nucleosomal DNA, especially, that affinity is actually helpful to reach the target site faster. So we wanted to know why this is so, because this is kind of contradiction to the present hypothesis, and we try to investigate into more detail. And we thought probably this is because of the dynamics of this nucleosomal DNA that can wrap and unwrap. So what we found is that the first plot, if you see on the top left, is that how the time changes if the nucleosomal opening rate changes. So basically, if the nucleosomal opening rate changes, the time actually goes down, and that is obvious because when the nucleosome opens fast, so basically the transcription factor, the pioneer transcription factor especially, it sits on the DNA and it waits when it will be opened. As soon as the DNA is open, it starts its journey towards its target site and it reaches faster. So if the waiting time reduces, that means the nucleosomal opening rate increases. So if the waiting time reduces, the search time will go down, and that is what we can see here. But if you see on the top right panel, that is for the non-pioneer factor. And in case of non-pioneer factor, if you change the nucleosomal opening rate, what you found is that completely different kind of shape, like it's a U-shaped plot. So what happens is that, so on for the non-pioneer factors, they do not really have a very high affinity for the DNA. So when they sits on the DNA, and if the DNA opens up, it can actually kicks off the protein from the DNA surface, right? So if you increase the rate, so basically the kicking of the from the surfaces increases, and thereby every time it goes back into the solution and start afresh. So every time if it has to start afresh, it will take a longer time to reach its target site. So when the K open is very high, it takes longer to reach. When K open is very small, that is 10 to the power minus 4 or so, what happens is that the protein can actually ends up bound in a wrong, trapped state. So basically, when it is trapped, then it is extremely necessary that someone gives it a kick to out of the trap, or it gets out of the trap somehow and then comes back. So basically, error correction is necessary, right? Which can kick the protein out of its trapped site and helps it to find, okay, you should start afresh. So that is not there when the nucleosomal opening rate is very slow, and then the corresponding time is very high. But when we found that when the nucleosomal rate is optimal or intermediate, you can have a fastest, that means the lowest target search time. So that is the fastest search process. Similarly, if you see in terms of pioneer factor, in pioneer factor, the protein gets out of the DNA surface when there is a dissociation happens, right? So in that case, the K off is the case. And we again get a similar kind of U-shaped structure. So in this case, what is interesting is that if you see the K on different for this, these three different colors are for three different K on values. And as the K on value increases, the basically minima shifts towards higher K off, right? So that means there is a kind of correspondence that if K on increases, K off also increases. And we try to look into this a bit detail, and what we found is that there is a novel mechanism which we call dissociation compensation mechanism. That means on transcription, pioneer transcription factor, you have this property that its K on and K off rates are comparable. If the association is very slow, then its dissociation rate is compensated such that the protein gets enough time to scan the DNA. If its association is very fast, its dissociation rate is also very fast, such that the protein should not get trapped into any site. Once it gets trapped, immediately it will be out, and the corresponding dissociation rate will kick it out such that it can start afresh and finally reaches. So basically there is a correspondence in these two directions, and this is called a dissociation compensation mechanism, and because of which we found that the pioneer factors are the one class of transcription factors which can really invade the nucleosome, and non-factors cannot invade because of this issue. So this is what we have recently found out. I will show you another journey of protein, but this time on a different track. So so far what we have shown is on a double-stranded DNA. Now I'll show you on a single-stranded DNA track. So this is a protein. There are 5 domains of this protein, and this protein is called RPA protein. That means the replication protein A. So this protein is extremely important, and what happens is that when you have DNA double-stranded, at the time of DNA replication, DNA basically unwraps, and a single-stranded DNA you can see. So the single-stranded DNA are extremely vulnerable, chemically degradable easily. So this RPA protein kinds of binds with the DNA to provide with stability such that the DNA doesn't degrade or doesn't form a. Two interesting observation was there experimentally. One is that if the DNA, so one amazing thing is that this same RPA protein binds with DNA of different length to result into different DNA metabolic processes. That is amazing. So for example, if the same RPA protein binds with a long, then it works in DNA replication. But if the same RPA binds with a short ssDNA, it works in a DNA repair, damage repair process. So basically the same intermediate, same things, but if the length of the DNA intermediate changes, it works differently. So we tried to understand why this is so. So interestingly, people have tried to crystallize the structure with the long DNA and the short DNA. And what they found is exactly the same kind of structure, this kind of structure, right? For both the long and short DNA. And actually they could crystallize only 25 nucleotide single-stranded DNA and not beyond that. So that means the rest part is extremely flexible and people couldn't identify its physical, its existence. So we created a model and we tested it on many single-stranded DNA and protein complexes, and we carefully looked into that, and we found the crystal structures, and then we tested on the same protein. So what we found is that on the left-hand side, you have a DNA, short DNA and the protein. So this protein is kind of extremely big and the DNA diffuses on the protein, right? And you get this kind of horseshoe shaped structure. And correspondingly, if you see, so what we have, we basically, the whole length of the DNA, we divide into two parts. The left-hand side, we named it as arm 1 and the right-hand side as arm 2. So when you construct a free energy diagram using this context of arm 1 and arm 2, the left-hand side, it shows that there is a pronounced energy minima. This energy minima, which is H25, is actually represent this U-shaped kind of structure. Now, if you see the same thing on the long DNA molecule, long ssDNA molecule, we clearly see two different energy minima. One we name as L62. In this case, this is the first time we have identified that the DNA is actually the arm 1, it forms lots of contact with the protein surface, but the arm 2 remains roughly flanking on the DNA surface, on the protein surface. And this assumes kind of linear structure, linear shape, and this state immediately turns into another slightly more stable state, which we call H62, in which you get this, again, this U-shaped structure. So this binding is quite different. We tried to find out all the corresponding, interactions that are governing this binding, and we found many more details. But what we are more interested in this case, that how actually DNA diffuses on this kind of protein. So this is for the short protein. The left-hand side top, this black diagram. If you see, basically what happens that when you have a protein surface, this DNA forms a kind of small bulges. The bulge are basically few nucleotides are forming a balloon-like shape on the protein surface. And over the time, this balloon moves, right? So if you see the lower panel, we have shown the movement of these bulges from one end to another end with at different time. And so actually, if you see the bulge on the top panel, which is named as DT25, you can see that this circle actually shows that the bulge actually moves. So there is the nucleotide indexes changes with time, and the bulge actually moves with time. So that means on a shorter DNA, the DNA moves through bulge formation on this kind of protein surface. So this is the kind of situation you have, that when the protein, when the DNA binds, see the arm 1 is bound, and then this part, it forms bulges and coming towards this, the right hand side, right? And to make kind of U-shaped like structure. So this is the binding of the single-stranded DNA, but short one. But if you see the long one, this is amazing, because in the arm 1, you see this, the indexes, the bulge indexes are roughly static in this range. More importantly, when you see the arm 2 context, yes.
Dr. Arnab, yeah, I think you please conclude, I think, now.
Right, this is the last, I think, one slide, I'll conclude. Yes. Right. So what we found is that on the arm 2, that there is no contact for longer time, and then the bulge forms, but they are more static. So basically, that means that the bulge on the longer DNA, that doesn't diffuse. So what we found is that there is a corresponding, for a longer DNA, you see a fly casting kind of mechanism where the arm1 contacts are formed to some extent, and then suddenly the arm2 contact formation started. This is basically because of the longer DNA length. There is a fly casting which senses the protein surface, the charges, and it immediately falls on the protein so that the contact formation is much faster. And that is the way that although the longer ssDNA are short-lived, but they can immediately fall on the protein surface to form a stable complex. So these are the conclusions that I've already mentioned, that the mechanism of this formation of for the small ssDNA, short ssDNA, it goes through repetition dynamics where the bulge moves. In longer DNA, it is through a cooperative fashion, which I said already. So this is the way, and we are enjoying our journey, and these work are mainly done by these two students, Anupam and Pinky, and I again thank you for inviting me, and I'll be happy if there is any question.
Thank you very much, Dr. Arnab. Such a fundamental research on understanding the protein transport on DNA tracks. Thank you very much. Probably if there is any question, one question somebody can say. Okay, there is a comment. There is a question by Dr. Dheeraj Sinha. What is the most critical parameter for specific in DNA protein interaction? So this is one question. Maybe you can shortly answer.
Right. So major contribution comes from electrostatic interaction, and apart from that, couple of more interaction. One is hydrogen bonding is very important, and the second one is base and the aromatic interactions, which happens between the nucleotides and the aromatic bases. So these three components are extremely important.
Okay, thank you very much, Dr. Arnab. And now we have come to the conclusion of this session, and I thank all the speakers for their presentation, all the participants, all the delegates for listening the talks, and also organizers for giving me the opportunity to chair this session. Thank you very much.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir.
Thank you.
Now we are going to start oral presentations, and in this series, for the first oral presentation, I invite Jyotsna Jaiswal. Jyotsna Jaiswal. Jyotsna, are you here? Jyotsna hai ki nahi? Jyotsna?
Hello.
Hello.
Ma'am.
Yeah, Jyotsna, you can present. You just start sharing your screen.
Ma'am, I can't see option here to share a screen.
Okay, just a wait. Start. Option is there, Jyotsna, in the right side, you see.
It is in the central lower corner of the screen of the Zoom app, the central lower corner, immediately in the center, lower part.
God, I can't see.
Beside chat, it's written share screen.
Yes, sir. Yes, sir. I can see. I can see.
Okay.
Hello.
Yeah, Jyotsna, we can see your screen now. Full screen, please.
One sec, ma'am. Ma'am, can I start?
Yeah, I'm giving you 5 minutes
for
your presentation and 2 minutes for question answers.
Okay, ma'am.
Okay, you can start. Okay, your voice is audible. You just start.
Okay, ma'am. Good evening, everyone. I'm Jyotsna Jaiswal from University of Allahabad, and my topic of presentation is antioxidative and antimicrobial properties of Argemone mexicana. Here I'm going to describe background of the plant species I have worked with. Argemone mexicana, despite of having many toxic effects, it holds a special place for medicinal benefits, which are being exploited since ages. Argemone mexicana contain various chemical compounds such as alkaloid, flavonoid, and terpenoids, which are being responsible for medicinal properties. Some workers have reported that leaves and seeds of Argemone mexicana have significant antimicrobial activity. The essential oil of Argemone mexicana is also found active against some microbial species. Here are my objectives. I prepared preparation of extracts of plant leaf and fruit of Argemone mexicana in different solvents. Spectrophotometric analysis of extract for presence of different phytoconstituents, assessment of redox potential of extract using DPPH assay, evaluation of antimicrobial potential using diffusion methods. In result section, we have prepared the extract of leaves and fruit of Argemone mexicana in different solvents like hexane, ethyl acetate, methanol, and water in increasing order of their polarity at high temperature equivalent to their boiling point of respective solvent using Soxhlet apparatus. The solvents, the extracts were analyzed spectrophotometrically to detect the presence of different phytoconstituents. The spectrophotometric analysis indicated that the presence of flavonoid pheophytin A and unsaturated carbonyl compound in various extracts of different part of the plant. Total flavonoid content in these plant extract range between 10 to 1150 μg quercetin equivalent per mg. Aqueous extract of leaf extract of Argemone mexicana displayed maximum free radical scavenging activity. Determination of IC50 of different extracts. IC50, the half maximal inhibitory concentration, is a measure of potential of substance in inhibiting a specific biological and biochemical function. DPPH assay of Argemone mexicana plant extract displayed potential free radical scavenging activity. Aqueous leaf extract of Argemone mexicana shows considerable IC50 value. Antimicrobial potential of Argemone mexicana leaf extract prepared in ethyl acetate and aqueous medium and fruit extract prepared in methanol shown notable inhibition antimicrobial growth. The inhibition zone for Citrobacter is 8 mm, 10 mm, 10 mm, and 11 mm for the concentrations of 50 mg per ml, 100 mg per ml, and 200 mg per ml, 300 mg per ml respectively. For fruit extract prepared in methanol medium, the inhibition zone of standard is 13 mm at 5 mg per ml concentration. The inhibition zone for Salmonella typhi is 6 mm, 12 mm, 13 mm, and 15 mm for concentration of 50 mg per ml, 100 mg per ml, 200 mg per ml, and 300 mg per ml respectively. For the leaf extract prepared in aqueous medium, the inhibition zone for the standard here is 16 mm at 5 mg per ml concentration. In the conclusion section, I would conclude with the results of all the experiments. The spectrophotometric analysis indicated presence of flavonoid pheophytin A in most of the plant extracts. Argemone mexicana leaf prepared in aqueous medium shown considerable inhibition value. Antimicrobial potential of Argemone mexicana leaf extract prepared in aqueous medium and fruit extract prepared in methanol have shown maximum inhibitory concentrations against their respective bacteria. I would like to acknowledge my supervisor, Professor Bichin Sharma, for his guidance and support, and my colleagues. Thank you.
Thank you, Jyotsna, for completing your talk in stipulated time.
Thank you, ma'am.
Now it's open for question and answers. Question and answers is if there is any question. Audience, it's open to you. Now, if there is no queries, then now we'll move to the second talk. For that, I'll invite Manoj Kumar for presenting talk for the title Heterosis Analysis in Indian Mustard under Various Nitrogen Levels. Manoj Kumar.
Hello, good afternoon, ma'am.
Yeah, Manoj, you can start your presentation.
Actually, my laptop is hanged, so it's not working. Sorry for that.
So will you present from mobile or you will not present?
I'll present from the laptop, but could you please give me the time?
Okay, if Sunita is there, we can move to third talk.
Yes, the third talk, and then go back to him.
Yes, ma'am.
Sunita, are you here?
Yes, ma'am.
Okay, Sunita, can you give you are ready? You can give your presentation.
Yes.
Okay, you share your screen and you start. Sunita, there is a 5 minutes for your presentation and 2 minutes for question answers.
Yes, ma'am. Is my screen is visible to you?
Yeah.
Okay, ma'am.
Sunita, your slides.
Yes. My slides. Yes, ma'am.
Yeah. That's okay.
A warm good evening to all of you. Thank you, Konyaps, for providing this platform to me. Here I am presenting my work dealing with the identification characterization of RV0707 as a potential vaccine candidate in Mycobacterium tuberculosis using in silico tools. I am working under the supervision of Professor Pratyush Shukla and the co-supervision of Professor Yogendra Singh in the Department of Microbiology, MD Rohtak. Starts with the introduction. The Mycobacterium tuberculosis is a deadly pathogen in the history of humankind, and it infects around 1.5 billion people and kills 1.3 million people every year. It consists of around 4173 genes in its genome, which is around 4 mega base pair. And here we are working on the DNA binding proteins of Mycobacterium tuberculosis, as they are responsible for different cellular process like DNA repair, replication, recombination, and consequently enhance bacterial growth and survival. So here we selected RV0707 as a DNA binding protein, and we work on its epitopes to predict as a potential vaccine candidate. Now here is methodology. We have retrieved the sequence of RV0707 through microbrowser database. It is a specific database for mycobacterial species. Then we predict it as a DNA binding protein with the help of a DNA binding software. Afterwards, the antigen, allergen, and the toxin property of that protein was predicted with the help of various softwares. Further, it was subjected for epitope prediction, in which the B cell epitope was predicted by ABC Pred server and T cell epitope was predicted by ProBred. Additionally, the 3D model of the protein and the epitope was built by the iTaser and PrepFold. After that, the docking study was performed to confirm its validation as a potential vaccine candidate. Now results. The RV0707 is a potential DNA binding protein with a DNA binding score of 3.566. The DNA binding probability score is 0.9725. The antigen score is 0.8755. It is predicted as non-allergen. It is predicted as an antigenic protein with a score of 0.979271, and it is also predicted as a non-toxin protein. The table 2 depicts the epitope sequence, the B cell epitope sequence, which is predicted by the ABC Pred. The ABC Pred score is 0.9, the cluster score is 26, the T cell epitope is, the sequence of T cell epitope is also shown below. The figure 1 depicts the 3D model of the protein, the RV0707, which is made by the iTaser. It was also validated through Ramachandran plot. After that, the protein and the epitope was docked, which is depicted in the figure 2 with the human HLA allele A. Now conclusion. In conclusion, RV0707 was predicted as a putative DNA binding protein of Mycobacterium tuberculosis. that could offer a novel idea to design novel therapies against TB. The study was performed to predict epitope of RV0707. The epitope was predicted on the basis of different properties like allergenicity, antigenicity, epitope mapping, and molecular docking. The epitope predicted from RV0707 may also trigger the B cell and T cell mediated immunity response. The assessment of the predicted epitope is extremely acceptable to prove them as an immunogen and potential vaccine candidate against TB. Now I acknowledge MT University Rohtak, University of Delhi, University Research Scholarship Rohtak, UGC New Delhi, and also the CONIOPS. Here is the references, and thank you for listening. Thank you.
Mute.
Thank you, Sunita.
Thank you, ma'am.
Now it's open for discussion. Okay, if there is no more question, then I'll move to the next talk. That is, Manoj, are you ready?
Next one after this.
Okay,
please.
okay, Manoj. Navneet Kaur.
Yes, ma'am.
Are you ready for the presentation?
Yes, ma'am, I'm ready.
Okay. Now I am inviting Navneet for presenting her oral presentation. That is, yeah, your presentation was previous day, but because of some problem, you didn't.
Yes, ma'am.
No, Navneet, you just share your screen and start.
Okay. Ma'am, is the screen visible?
Yeah.
Okay, ma'am.
The screen is clear.
A very good afternoon to all. I'm Navneet Kaur from STOS Jammu, and the topic of my presentation is bioactive compounds from Syzygium cumini, that is jamun, fruit exhibits anti-cancer efficiency. First of all, what is cancer? I'll give a little bit of a description. That is, cancer is a general term used to describe a group of diseases that are characterized by specific genetic mutations and that give rise to a loss of control over critical cellular functions. Cancer cells differ from normal cells in many ways that allow them to grow out of control and become invasive. It forms when genes within normal cells are mutated. The objective of the studies are determination of in vitro cytotoxic effect of root extract against various human cancer cell lines. The next one, bioassay guided fractionation of active extract and further their evaluation. And the last is characterization of most active fractions and ingredients. And the fruit that was taken was Syzygium cumini, that is locally named as jamun. Its methanolic extraction was done, and then fractions with different solvents like hexane, chloroform, and butanol was done. And the isolation of mainly 3 compounds was done from the active chloroform fraction. And the methodology is here. Am I audible? Ma'am, am I audible?
Yes, you are. Please proceed.
Okay. The methodology is extraction of root material was done with the methanol solvent, and then evaluation of extracts was done for in vitro anti-cancer efficiency using specifically SRB assay, and then fractionation of active extract with N-hexane, chloroform, butanol, and further evaluation of fractions was done, and then isolation of bioactive compounds using column chromatography, and their characterization was done via NMR and mass spectroscopy. The results demonstrate that specifically the methanolic extract showed activity mainly against 3 of the cancer cell lines. That was HCT116, A549, that is lung, and prostate, that is PC3. And further, their fractions, that is hexane and chloroform fraction, mainly showed maximum activity against maximum number of cell lines. Based on this activity, biomolecules were isolated, mainly oleanolic acid, gallic acid, and quercetin from the active chloroform fraction, and then the activity of these compounds was done on the same number of cancer cell lines. And it was found that in case of oleanolic acid, The compound was found to be active more on colon, that is HCT116, and on MDMB231, that is breast cancer cell line, with the IC50 value of 18.78, very less in case of A549, and 40.51 micromolar in case of colon cancer cell line, that is SW620. The second compound, that gallic acid isolated, was again screened against a number of cell lines, and it was found to be very much active on the specifically colon cancer cell lines, that is HCT116 and SW620, with the IC50 value of 54.26 micromolar and 54.5 micromolar in case of SW620. The third compound, that is quercetin, showed maximum number of growth inhibition against maximum number of cancer cell lines, as you can see here, with the IC50 values of 23.25.36 in lung cancer, and very less also in HCT116, in SW620 and MIA-PACA. So here I would like to conclude with my presentation that S-cumini, that is jamun extract, was found to be active on 3 of the cancer cell lines mainly, that was colon origin, from lung origin, and from prostate. And mainly 3 compounds, that is olenolic acid, gallic acid, and quercetin, were isolated from the active chloroform fraction. And specifically quercetin showed potent in vitro cytotoxicity, and that could be developed further as a potential anti-cancer agent. Thank you.
Thank you, Navneet.
Welcome, ma'am.
Now, if there is any question, it's open for discussion. Okay, now we are moving to the next talk. That is Manoj, are you ready?
Ma'am, actually, I do have some issues.
Okay, no problem, no problem.
Okay,
Now, in the last presentation, now it's last. After that, you should be ready, okay? Next, Priyanka. Na? Priyanka, are you ready for the presentation? Priyanka Tiwari? Priyanka, are you here? Then Nikita? Now one more student is there, Monica Yadav. Monica. So Monica, no one is there, okay? Manoj, now it's time for you. If you are ready, then we'll go for your presentation. Otherwise,
Monica has joined.
Monica. Monica has joined. Okay, Monica. Monica Yadav, are you ready for the presentation?
Madam Priyanka Tiwari is also there. She's writing a message. We, for some reason, cannot hear Monica. Monica, we cannot hear you.
Priyanka Tiwari, are you ready for the presentation? Priyanka?
I think she has joined with the wrong link. Priyanka, please use the link which was sent to you for the presentation, not the general registration link. Otherwise, you will not be able to present here. Madam, back to you. She's here. Back to you, madam.
Okay, Priyanka Tiwari? It's Monica Yadav. Okay, Monica Yadav has joined. Monica, are you ready for the presentation? Monica, you are not audible. We can see your screen, but there is no voice. मोनिका की आवाज नहीं है ना अनम्यूट वाली प्रॉब्लम तो नहीं है वो देख लेते हैं का Monica, we can see your screen, but you are not audible. Monica, we can't hear you.
Madam, now Priyanka has taken over the screen.
Priyanka. Okay, Priyanka. Priyanka Tiwari. Priyanka Tiwari का screen तो Priyanka, you are also not audible. Chat box. Priyanka, I think you are sharing your screen, but we can't hear you. She's moving on. She's saying, she's presenting even, but we are not hearing. तो क्या किया जाए? हम लोग की आवाज तो Then.
Yes, ma'am, we can hear each other, but for some reason we cannot hear Monica nor Priyanka.
Yes,
for
sir.
some reason. That is very
Yes,
strange.
sir.
Maybe
Sir,
their headphones, they should change their headphones.
She has presented now last slide also. Thank you.
Maybe we can go back to Manoj if Manoj is ready, ma'am.
Manoj, it's last call for you. Manoj, are you ready?
Actually, I'm trying to, but it's not
Manoj,
opening. No.
Manoj, your voice is clear.
Can I present orally? Actually, I am not able to display my slides.
Okay. We are giving you time. You just conclude in 3 minutes. Okay. You present your work. Without presentation, you present. No, you have started screen. Yeah, we can see your screen now.
Ma'am, that is Monica. That is Monica.
Okay, Monica. Sir, एक की आवाज है, एक की screen है.
मुझे लग रहा है मनोज जी मोनिका का presentation देंगे.
Okay, this is also good.
No, I think, ma'am, if the other two is not audible, you can do with Manoj orally without a presentation, and
yes,
in the
sir.
meantime,
Yes,
the
sir.
other two ladies can figure out why their audio is not working or change their laptop or something. इस तरह करा लीजिए, मनोज जी से verbal करा लीजिए.
जी. Manoj? Sir, I think Monica is not hearing us also.
I have written to her that we cannot hear you. Please check your speakers. I've written both to Priyanka and to Monica. Madam, if you proceed with Manoj, we can at least hear him what he has to say.
Yes, sir. Yes. Manoj, you start your presentation without sharing your screen. You start your presentation.
Unmute your mic, Manoj.
Okay. Good evening, all. My study title was the heterosis analysis under various nitrogen levels in Indian mustard Brassica gentiana, and I used 10 parents based upon the genetic diversity analysis. And after the genetic diversity analysis, we selected we selected 10 parents, and from the 10 parents, we produced 45 crosses of the mustard, and we evaluated those crosses under various nitrogen levels. Like there were 3 nitrogen levels. One was control condition, second was 75 kg nitrogen hectare per condition, and third was the 150 kg nitrogen per hectare condition. And when we analyzed the data pertaining to the crosses, and it came to our finding that the cross Jumka into RGN3 was the best performing cross among the different crosses, and Jumka parent was one of the best performing parent among the 10 parents. And there were 2 other crosses which also performed better. They were RGN7 into MGN30, and one was Jumka and Varuna 30. And based upon the observation which we have observed, so we can use these 3 crosses. Like Jumka into RGN30 and Jumka into
Madam, Manoj's mic is mute again.
Am I audible?
Yeah.
Okay, ma'am. Okay, so based upon these results, so we can use these two crosses for the coming hybrids. So that would be the better, because in Brassica Genci or Indian mustard, there are very limited scope for the hybrids. So we can use these hybrids for the different nitrogen conditions, and that could be better for the farmers, farming community, and for other researchers also. Thank you.
Okay, Manoj. Okay. Thank you, Manoj.
Thank you
for your presentation. Now, Monica. Monica is still here. So, Monica, we are giving last call to you. Monica, if you are ready, you can start your presentation. Otherwise, we'll move to the second one. Last one, okay. Monica? Monica or Priyanka, if anyone is present? No. Then we'll move to the...
Madam, both are present, but apparently their audio is not audible.
Sir, Monica, mic is mute also.
Mm-hmm. Monica, please unmute your mic. On the lower left corner, there's the mute, unmute button. Please click on unmute. Yes. Can you say something? Monica, can you say something? Madam, I think there's some audio hardware problem with your laptop, okay. I think.
So with this, we are ending the oral presentation here. Now I am handing over the charge to Dr. Vijay Tripathi for rest of the conference. Okay, Dr. Vijay Tripathi.
Thank you, Dr. Mishra. Now this is time for the vote of thanks and
Dr.
conference.
Dr. Tripathi.
So good evening to all the distinguished guests, dignitaries, panelists, speakers, invited guests, ladies and gentlemen. It is my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of CONIAPS 26 and the entire JIP team, let me call it fraternity here together and on my own behalf and extend a very heartily thanks and would like to extend my sincere gratitude to the panelists. And speakers for sharing their expertise with us and showed a better way to discuss the current scenario in the field of biotechnology, bioinformatics, and biochemistry to eradicate the problem faced by humanity, especially in human health. I took note with great interest of your valuable interventions. I would like to take this opportunity to extend my heartfelt gratitude to Most Reverend Professor Dr. Rajendra B. Lal, Vice Chancellor, SUADS, to give us permission and support to conduct the international conference. I express my gratitude to Professor Dr. A.K. Lawrence, Pro Vice Chancellor, Academic Affairs and Quality Assurance, Professor Reverend Sarvajit Herbert,
Pro Vice Chancellor, Planning, Monitoring and Development, Professor Dr. S. Bilal, Pro Vice Chancellor, Administration, Professor Dr. Robin L. Prasad, Registrar, for their time-to-time support throughout the conference. I am very grateful to Engineer C.J. Wesley, Joint Registrar, for his valuable support, especially providing technical support to conduct this conference in online mode. This conference would not have been possible without you, sir. I would like to extend my gratitude to Professor P.N. Pandey, General Secretary, International Academy of Physical Sciences, Prayagraj, for their valuable insight for conducting this conference. I am also grateful to Professor Professor Dr. I.R. Jonathan Elal, Dean Jib, and Director of IPC for the perfect guidance he has extended to all of us at CONIAPS 26. I express my gratitude to Professor Dr. Kapil Lawrence, Head Department of Molecular and Cellular Engineering, for being there with our entire team in organizing this conference. The aim of this conference were high as they examined many challenges, resources, depletion, land management, food waste, and human health, combating with the problems with the innovation idea brought up by the young minds of our nation. Finally, I express my gratitude to my two students, Mr. Prashant Tiwari and Mr. Vibhav Srivastava, who were my backbone for this entire team. I have been fortunate enough to be backed up these two highly motivated and dedicated students of JIB who know their job and are result oriented. I cannot thank everyone enough for their involvement and their willingness to take on the completion of tasks beyond their comfort zones. And I would like to thank all of you for your active participation and open dialogue. I look forward for continuing beyond this event, the very stimulating and instructive exchange of ideas we had today. Thank you. Now, I would like to call Dr. Pragati Mishra for the summary of the conference. Dr. Pragati Mishra.
Thank you, Dr. Tripathi.
Madam, just one query. The two students who were not able to present, their abstract will be published in the conference book, right?
Yes, sir. It's already published.
Thank you, ma'am. Thank you.
Okay, sir. So now I'm going to give the technical report of the 3 days conference. The 3 days international conference, CONIAPS 26, commenced on 18 December 2020 through online mode via Zoom. The conference is organized by the Department of Molecular and Cellular Engineering, Jacob Institute of Biotechnology and Bioengineering, Sam Higginbotham University of Agriculture, Technology and Sciences, Prayagraj, in collaboration with International Academy of Physical Sciences, Prayagraj, India, and Mahatma Gandhi University, Kottayam, Kerala. Professor Kapil Lawrence, Head, Department of Molecular and Cellular Engineering, welcomed the participant on the first day, and which was followed by the address from the engineer C.J. Wesley, Joint Registrar, who gave the introduction of the university, highlighting its academic as well as research profile. The introduction of the conference was given by the organizing secretary, Dr. Vijay Tripathi, which was followed by the address from the convener, Professor Dr. Jonathan Elal, Dean Jacob Institute of Biotechnology and Bioengineering, Schwartz, who emphasized on the significance of international conference on recent advances in biotechnology, bioinformatics, and biochemistry. Professor Lal called it an amazing conference for it, as it will help us know biotechnology and allied sciences, understand it, and learn to grow with it in present pandemic world. Now, technical session of day 1 was chaired by Professor M.P. Singh, University of Allahabad, which included the first day technical session included 4 lectures from scientists and academicians. First lecture was delivered by Dr. Dennis Hogan, Executive Director, European Alliance for Personalized Medicine and Co-Editor-in-Chief, Public Health Genomics, Belgium. During his talk, he introduced the audience about the importance and the benefits of personalized medicine, like customized disease prevention strategies, more effective drug delivery, reduced time cost, failure rate of pharmaceutical clinical trials, etc. in the present pandemic world. Second lecture was delivered by Adesitrin, Institute of Soil, Water and Environmental Science, Agriculture Research Organization, Israel. His lecture was focused on how treated wastewater holds potential for the use in irrigation, suggesting new ecological and epidemiological insights on the contribution of treated wastewater irrigation to the potential dissemination of antibiotic resistance, necessitating the refinement of existing and future risk assessment practices. Dr. Dheeraj Sinha, third speaker, IHU Mediterranean Infection, Marseille, France, presented his talk on pangenome analysis of Streptococcus intermedius and how high-throughput genome sequencing methods are enabling in understanding the genetic landscape and diversity of bacteria, as well as their pathogenic role. Next lecture in this series was delivered by Professor Pratyush Shukla, Professor, School of Biotechnology, Banaras Hindu University, Varanasi, on futuristic ideas for biocomputational and systems biology approach in microbial biotechnology. The microbial biotechnology as an emerging due to innovative applications, and there are numerous new perceptions in microbial enzyme engineering which are gaining attention nowadays. After the invited lecture, oral presentations was presented by various researchers and students on the conference focal theme. Now on 19th, that is on the second day of the conference, Professor Rajiv Kant, Head, Department of Computational Biology and Bioinformatics, chaired the session. Secondly, the first lecture was delivered by Professor Syed A. Rizvi, Chairman, Institute of Professional Studies and Dean, Research and Development University of Allahabad, Prayagraj, India. He delivered his lecture on anti-aging strategies based on caloric restriction mimetics. He presented talk showed the anti-aging role of metformin on rats. Next lecture was delivered by Dr. Vishwanath Tiwari, Department of Biochemistry, Central University of Rajasthan, Ajmer, Rajasthan, India, and he was the second speaker. His talk was focused on multidisciplinary strategies for combating carbapenem resistant Acinetobacter baumannii. He presented that the conformational stability of the recombinant protein plays a vital role in retaining the function of beta-lactamase even under stress condition. His experimental and bioinformatics studies showed that carbapenem is effectively hydrolyzed by OXA51. Now, third lecture was delivered by Hemchandra Jha, Department of Biosciences and Biomedical Engineering, Indian Institute of Technology, Indore, India, on COVID-19 trends and recent updates. His talk provided us an insight about SARS-CoV-2 causative agent of current pandemic disease COVID-19. He also talked about the various aspects of COVID-19, which involves epidemiological studies, comorbidities associated with it, like neuro and gastric related complications, drug repurposing, and pharmacological approach for drug discovery. Next lecture was delivered by Professor Sarvas More, Institute for Public Health Genomics, Faculty of Health Medicine and Life Sciences, University of Maastricht, the Netherlands, was the fourth speaker. His talk was on vaginal microbiome composition, which can predict IVF failure with extremely high clinical accuracy. He gave us an insight about woman reproductive health line, how next generation sequencing and his new patented IS Pro technique is used for the microbiome analysis. Fifth speaker On second day was Dr. Anita Krishnan, ex-staff scientist, Department of Ophthalmology, Harvard Medical School, Massachusetts, USA. She delivered her talk on insight on gene therapies for ocular diseases. Her talk dealt about the mechanism of adenoviruses-mediated gene therapy for treating glaucoma. Dr. Sushil Mishra, last speaker on second day, Department of Pharmacy, University of Mississippi, USA, delivered lecture on computational glycoscience and drug designing. He spoke about the challenges that exist in the modeling of protein, carbohydrate recognition through computational approaches, and a number of case studies showing how well these techniques can be used to predict the change in binding affinity and specificity of proteins upon mutations. After the invited lectures, oral presentations were presented by different researchers and students on the conference theme. The third day of the conference, Professor P.W. Ramateke, ex-Dean Postgraduate Studies, Schwartz, chaired the third and final session of the conference, which included 4 invited lectures. First lecture was delivered by Dr. Dhruv Kumar, Associate Professor, Amity Institute of Molecular Medicine and Stem Cell Research, Amity University, India, on understanding the signaling regulations and mutational heterogeneity in cancer and cancer stem cells. He emphasized on important aspects in cancer metabolism and interdependence of autophagy in the survival and progression of cancer cells. Second lecture was delivered by Joost Andre, Faculty of Biosciences and Aquaculture, Nord University, Norway, on landscape genomics from understanding adaptive potential to biodiversity conservation and natural resource management. He presented an overview how they support biodiversity conservation and natural resource management. Lack of understanding of these issues has triggered the development of landscape genomics as a tool to quantify shared and unique adaptive responses across species within large and ecological diverse landscapes. He presented an overview of these developments and illustrated how they support biodiversity conservation and natural resource management. Professor Dr. Jonathan A. Lal, convener, CONIAPS 26, welcomed Dr. Sasmit Patra, Rajya Sabha member, Odisha, as a chief guest. Dr. Patra addressed the audience and talked about various policies matters in biotechnology, biochemistry, and bioinformatics. He emphasized on the importance of patent technology and the significance, what is the significance of raising funds in the research work on various topics of science and technology. He assured us that he will put the matter of the scientific matter in the parliament also. After that, the third and final lecture of the session was delivered by Dr. Arnab Bhattacharjee, Associate Professor, School of Computational and Integrative Sciences, Jawaharlal Nehru University, New Delhi. He presented his talk on how protein travels on DNA track. He discussed the physical basis of such fast and accurate target search mechanism of DNA binding proteins on both single and double-stranded DNA track. After that, the oral presentations were presented by various researchers and students of different schools. So this is all about the technical report of 3 days international conference. Now, now it's the most awaited event of the international conference. I'm going to announce the names of the winner, names of 3 days, this oral presenters. For this, first of all, I would like to thank all the participants who presented their research work as an oral presentation and helping make this conference a great success. So for announcing the name of the prize winners of this oral presentation, in this series, first prize of oral presentation goes to Dr. Jaydev Sharma and Dr. Ajit Kumar of MD University, Rohtak. He delivered talk on designing inhibitors for COVID-19 main proteases and their ADMET analysis. I want applause for this. Now, second prize for this goes to Shweta Manhas, Vikas Sharma, Diksha Kaul and Asha Chaube team for her talk on optimization of L-asparaginase obtained from Enterobacter ubeaceae using response surface methodology, isolated and characterized from black gram rhizosphere soil sample of J&K. Third winner in oral presentation series, third prize goes to Sunita Chetkar, Chandrakeshavam, Yogendra Singh, and Pratyush Shukla for her talk on identification and characterization of RV0707 as potential vaccine candidate in Mycobacterium tuberculosis using in silico tools. So this is all about the conference. Now I'm handing over to Vijay Tripathi, Dr. Vijay Tripathi, for concluding remarks. Thank you.
Thank you, ma'am. Now I would like to call Dr. Ipan Pikosi for the closing prayer. Dr. Ipan Kosi, sir.
Let us bow our heads in prayer. Lord, our Heavenly Father, we thank you for keeping us safe in your hands during this great time of pandemic and giving us an opportunity to praise your name once again, O Lord. O loving Lord, we come to the conclusion of this 3-day international conference. We thank you for your mercy and kindness for conducting this conference in a good way, Lord. We praise you for all the timely help given to us for successfully organizing this conference. Thank you for all the eminent speakers who had shared their profound knowledge. Bless them to delve more into the depths of the science and use them for the betterment of mankind. We ask your blessings on all the university authorities. Give them the wisdom to lead this university to greater heights. Lord, bless the organizers of this event. Strengthen them in all their endeavors. Lord, bless all the academicians, researchers, and students who participated in this conference. Help them to use this knowledge gained for the benefit of our society. We ask your blessings on all those who are in the front line of defense against this pandemic. Shower your blessings abundantly on them and keep them safe in your hands, O Lord. We ask your blessings on each one of us. Keep us safe in thy hands, O Lord. We ask this prayer in the mighty name of our Lord and Savior Jesus Christ. Amen.
Thank you, sir. Thank you to all for the joining this conference. So now we end this conference, and now we will meet again for next international conference. I hope so. Okay, thank you very much. Good night. Thanks for the joining. Good night, everybody.